Lars, can you listen us? Can you check that you have
Lars, can you do you have the translation services, Lars? Can you hear the translation services?
You can't start your video because the host has stopped it. So if you can allow me to share my video.
Yeah, I think that Julian will give to us.
Ángel, por favor, poner como
Ángel, please enable Lars as co-host.
Yeah, I tried to open it, but let's see here.
Listo.
Gracias. Listo, Lars.
Lars, you have already been enabled as co-host. Now you can share it. Hi, how are you doing, Lars?
Yes, hello.
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 Is this the NSDF meeting?
Bueno, ya son 60, casi cerca de 60 personas conectadas en YouTube. La ventaja de YouTube es que pueden regresar.
We have 60 people who have already logged in YouTube. The advantage of YouTube is that they can start if they want, whenever they want. So if you all agree, Marcos and Rosa Maria, I would like to give you the floor to make the opening remarks. Thank you so much. We are ready then. You have the floor now. Thank you very much. I would like to thank you all for being with us. I would like to thank all the speakers for their support, all the panelists coming from different sides of the world. According to our program, first we have the opening remarks by the director of the Institute. My name is Julian Carrillo. I'm a researcher of the Engineering Institute of the National University, and I would like to give the floor to Rosa Maria Ramirez, who is the director of the Institute. Thank you so much, Dr. Julian Carrillo. Good morning. I would like to welcome you all to the first virtual workshop on the potential of epidemiology of water of WBE in Latin America, an approach to antimicrobial resistance that will be held throughout 2 days. First, I would like to thank for the invitation of the Red Panamericana de Epidemiología Ambiental in order to open this event. And I would like to thank Dr. Julian Carrillo, the co-leader of this network, for this kind invitation. He's a researcher of our Engineering Institute. And I would also like to congratulate the Inter-American Network of Environmental Epidemiology for organizing such a workshop because this is a tool that has been used in the last decades in order to know the spreading of diseases. Because of the current pandemic, its use in the analysis of wastewater has increased in an exponential way in order to foresee outbreaks of this disease throughout the world in the Latin American region, in spite of the fact that there are several pilot projects in different countries on the detection of the SARS-CoV-2 virus. There is a limitation in the use of this tool by the authorities of the health sector. In Mexico, the Engineering Institute of the National University has been the first one to apply this tool for projects which have already been applied in several cities, as large as Mexico City, Querétaro, and the Engineering Institute is actively participating in the network of the Panacea International Civil Association, where Dr. Julian Carrillo participates as well as other academicians. And this has resulted in very important activities, at least in Mexico. Finally, I hope that the results to be obtained in the development of this workshop with the participation of experts on the topic from different countries will be useful in order to identify, strengthen different points for study, collaboration, and have a greater impact, namely to solve problems which are as important as those associated to pathogenic diseases, which unfortunately we are living now. Thank you so much for allowing me to make these opening remarks. I wish you great success as well. And now I would like to give the floor again to Dr. Julian Carrillo. Dr. Carrillo. Thank you so much, Rosa Maria, for the opening remarks to this workshop. Before giving the floor to Marcos Quintela, who will be describing in greater detail the Panacea Network, I would like to thank the Institute for its support, for the institutional support, the technical part, the dissemination part, and the support of the academic unit in Querétaro that has allowed us to organize this workshop, and of course, all the members of the Panacea Network, which is a joint organization of multiple researchers and academicians, people associated to research institutions of several countries. Dr. Marcos, you have the floor now so that you can elaborate on this Panacea Network. Thank you so much, Dr. Rosa Maria, Dr. Castillon, thank you so much. It is an honor to welcome you to this event. From the Panacea Network, I would like to thank you at the National University who organized this. Julian, thank you so much. For those of you who do not know what Panacea is, a network that was created as a response for the lack of a collaboration arena in the region, mainly during the time of the pandemic. This is a space of knowledge that was created in North America and Europe, showing their efficacy and existing many of these type of cases in the pandemic. Our objective is being a driving force for all the research which are at the domestic and international level, giving a boost to the region and promote the internationalization of this knowledge. Today we have members and institutions of at least 12 Latin American countries in strategic alliances with European institutions that provide us with funds for the institution. We have several types of works in different types of projects. Domestic projects in Latin America with powerful groups, and we all know that there are groups with less resources to which we also have to provide support. We were born as a response to the coronavirus pandemic, but our objective is the implementation of this tool to fight against other diseases as well as against other type of problems. Today we have a panel of experts with which we want to understand what is happening today in Latin America regarding these issues and how it is that we can use this tool in order to start monitoring infectious diseases as well as the resistance. I would like to give the floor now to the panelists I hope that those of you who are not now in contact with Panacea, please contact us. This is our email, and you can log in through our website. Thank you very much, and I hope that you will enjoy this course. Thank you very much, Marcos. Then we are going to start. The moderators of the first session are Maria Inés Saloni and Dr. Aldo Guerrero. Maria Inés and Aldo, you have the floor now. Well, thank you so much. Thank you so much, Julian. Good morning, everybody in Good morning, Latin America. Good afternoon, those who have logged in from Europe. Welcome to this workshop. First, I would like to thank Dr. Julian Carrillo from the National University and Marcos Quintela of the Newcastle University and current president of Panacea for inviting me to participate at this workshop. Together with Maria Inés Sanoli, manager of the laboratory in Sao Paulo, Brazil, we are going both to act as moderators in this session that will have two different presentations. The first one, and I would like to introduce Dr. Bruno Gonzalez-On. He's a medical doctor right now. Dr. Gonzalez directs the antimicrobial resistance unit of the Potencial University in Madrid, Spain. He's working in molecular microbiology and ecology for microbial resistance. The title of his presentation is Problems of Antimicrobial Resistance and How We Can Use Wastewater Epidemiology. First, I would like to remind you that the presentation of Dr. Gonzalez that would last from 20 to 25 minutes. After that, we're going to have a 5 to 10 minute period for questions that you will be able to ask through the YouTube platform. Dr. Gonzalez, thank you so much for participation at this workshop. You have the floor now.
Very good. Well, thank you very much. I want to take advantage of this opportunity to thank you for the invitation and also congratulations for the initiative. I think that during the pandemic, we have developed a great number of tools with some difficulties, of course, as you know, in the university, in the Complutense University, but it's a very good idea to keep these tools, implement them, and maybe to extend them. And I think that in the specific case of antimicrobial resistance, to widen the network of vigilance for COVID, that would be very important and would give us very important and fruitful outcomes. I want to also say that this is very important because it's the resistance of antibiotics is also something that we have to address. So I'm going to share with you an introduction to antibiotics, to the current problem, and also some things that we have done with the wastewaters, and that could be helpful to give the first steps. Well, without further ado, as you can see, this is the land, our Earth, our planet looked from the moon. And focusing on problematic of microorganisms that live especially with the presence of water in the earth. This is the topic, so I think that it is affecting us a lot. So we can see the world population over the last 12 years and UN projection with a flat or plateau. And then we can see up to 2000, 20th century that has revolutioned the quantity of microorganisms that are resistant. So we are more or less 80,000 million people in Earth. And also, when we talk about the intestine of humans and every gram of content of every human being has many microorganisms and bacteria, as many as in each gram. For example, we have 8 million inhabitants, 8 million bacteria. So when we talk about who are the human beings that are important in the planet, are those that are able to colonize all the ecosystem, but there is not a single ecosystem that has been colonized by bacteria. And no, there is no a human being that has not been internally colonized by bacteria. So the champions of the life in the planet are bacteria. that colonize all these ecosystems of Earth, not pacifically, but also fighting against them. So they have emerged during all this time. We have bacteria that resist antibiotics and colonize an ecosystem. They reduce or get rid of competitors, and other bacteria have tried more or less during 4,000 years have colonized these ecosystems where these antibiotics were. So that means that the struggle, the microorganisms that produce antibiotics and bacteria that try to struggle or fight against these antibiotics is as old as the life in Earth. And therefore, it is intellectually myopia to think that using antibiotics, we're going to eliminate bacteria. We have been trained a lot with the genomics and many other things, and therefore, we won't be able to get rid of them. And we see that we have only through 100,000 years being in Earth. In fact, we have discovered mammoths with the genes of antibiotic resistance. In these antibiotic resistant genes are in the environment since always, I mean, forever. But let's start clinically since less than 100 years ago, thanks to these antibiotics that were discovered. There is this explosion or this burst of human beings in Earth, probably also with the potabilization of water, vaccines. I'm not going to discuss this, but really with all these factors together have resulted in us human beings being this species that is so important in our planet. And therefore, my personal opinion is that the highest discovery in the history of humanity is the discovery of antibiotics that had made possible the development of all modern medicine. We would not know the modern medicine if it wasn't for the discovery of antibiotics. This is also, for example, an instance, the National Transplant Organization, the best in the world, and all these surgeries and these transplants, but all the surgeries and therapy against cancer is only possible because the antibiotics are working. And more or less in the production of resistance to antibiotics, we start with Fleming that learned that out there in the ponds, in the environment is where bacteria produce antibiotics. So all the researchers in this planet started discovering all the families of antibiotics in the 70s and 80s. We had a super habit of antibiotics, and we came to the golden era of the antibiotic discovery time. And we have, of course, an excess of antibiotics. What happens is that the industry focuses on other types of molecules. Excuse me. And so focus on other types of molecules, and then we have for 30 years been using the same families of antibiotics. Therefore, today we have a high levels of resistance and very few new antibiotics in the market or under development. The idea is that the idea is that for the 2050 or before, the first cause of death in the world is going to be resistance to antibiotics, with 10 million of people a year in Earth, in our planet. And the worst thing is that also the consumption of antibiotics is increasing in all the world consistently. So it's not even, it's not steady, it's also increasing also because of the increase of human beings and the modernization of many countries. And I don't know if it is good or bad, but those antibiotics are also can be disseminated over the world very easily. And there hasn't been any antibiotic in the market that has not created a resistance, immediate resistance. And when I mention immediate, to the point that at present we have localized bacteria resistant to antibiotics that have not even been launched to the market. They are not in the market. You can see here, for example, antibiotics that in Europe are not launched, are not approved by EMA, but we already have microorganisms with resistance to these antibiotics that are not even launched in the market. This is one of the situations that we have around the world. And as we can see here, this increase of consumption of antibiotics is not only in developed countries, but also in the low-income countries, underdeveloped countries. We see the increasing the consumption of antibiotics. So there we have to pay more attention not only to the development of resistance, but also we have to prevent or be ahead, to prevent this resistance. So although I don't like this very much, I didn't not like this slide, but we use antibiotics in humans and in animals in all the ecosystems in Earth. And the truth is that most of these antibiotics in our patients, humans or animals, are not absorbed. Most of them are not absorbed by the patient, really go to the waste waters and go through the excretion means of animals and humans. The first patient with penicillin, the excretion was in the urine, left penicillin, and it came again and was injected again. This penicillin was purified and was injected because the amount of penicillin that we do not absorb really is something that, the same penicillin that was left from the patient, that was released from the patient, was injected again once purified. And the truth is that in spite of the fact that we know that antibiotics are a problem in terms of the resistance, we know that this antimicrobial resistance is out of control. I showed you the example of Europe, but also in Latin America. These are the latest data from Labra. And as we know, third generation cephalosporin, we have more than 50% in our countries have resistance to cephalosporin. And this is an antibiotic that we use intra-hospital, also carbapenems. And meropenems are the antibiotics of last resort that are used in hospitals. This is IV, and in some countries we have levels of resistance also of 50% to them, which is very important and leads to the mortalities that we know. Who dies? Well, the patients, anonymous patients, every day in our hospitals. They die in a hospital for an Acinetobacter baumannii resistant to colistin, or sepsis after an operation, or complications by Klebsiella pneumonia resistant to tricycline. Different bacteria pathologies that are different, but always that lead to death because a bacteria is as resistant to, resistant to antibiotics. In the past, we used to talk about multi-drug resistance. Now in all our hospitals, we have bacteria that are resistant to all the clinical options that we have available. So the resistance to antibiotics is the biggest problem that we have seen. And we heard this in 2016 by the UNO, and we will say, well, maybe this is because of the pandemic, this hasn't changed. But this is why, as in 2016, we are now in the WHO determining which antibiotics are these clinical antibiotics that we have to maintain exclusively for humans, and how we can manage all these antibiotics that we use in that are used in veterinary medicine. Therefore, we are making these types of lists, but always from the point of view, knowing what type of antimicrobials we can use. As I said to you before, we use antibiotics in human medicine, in animal medicine, and agriculture in many countries, and therefore you see in all these ecosystems we generate resistant microorganisms. How it is resistance created? Well, there are mutations, random mutations. However, when we use an antibiotic, we select this bacteria resistant to the antibiotics, and this bacteria also is impacting the ecosystem. The worst part is that the bacteria do not stay there. Only having this resistance. Once they are effective, these resistance genes are going to be transmitted to other bacteria. So this is what we call, a gene resistant to an antibiotic is transmitted through these genetic structures that we that we call plasmids. A bacteria could have a plasmid not only with a resistant gene to antibiotics, but could have several genes. It could transfer a bacteria that is sensitive to antibiotics, and all of a sudden is going to be multi-drug resistant or resistant to antibiotics in a single step in a couple of minutes. This is the power of the dissemination of antimicrobial resistance. That's why it's so important to take an eye on that. When we use antibiotics, we have antibiotics in our system. The important thing is not only to see what pathogens are resistant to antibiotics, salmonella, tuberculosis. It's also what genes of resistance they have, because really this is the reservoir in the ecosystem. If a patient is treated with antibiotic, every time that we prescribe an antibiotic, it's there in the intestine, and these resistance genes to antibiotics are going to be inside this ecosystem, and therefore we have this ecosystem that is a reservoir of resistant genes that are going to be transmitted to any pathogen bacteria that comes into this ecosystem. So how to preserve the action really of the antibiotics? Well, here is where we have this concept of we see the human health is intrinsically related with the health of animals and the health of the environment. It is really absurd to just look at these separately in silos. They are completely together. This is a joint, and the concept is not as modern as it looks. The concept, we knew, for example, that rabies that affected dogs, we had to vaccinate the dogs, eliminate the dogs of the eliminated rabies from the dogs to eliminate it from human beings. We have always known this relationship, animals, environment, human beings, but the concept of one head is exactly something that started in the year 2000. But why is not really applicable? Why is not implemented? Because when there are institutions that work in, they have different forms to work. For example, these institutions tend to avoid the problem. For example, when we have physicians, biologists, pharmacists, they start the blame game, So don't tell me as a veterinarian how to use the antibiotics if you are a pediatrician and you prescribe it to children. Well, the pediatrician says, well, okay, I do it because I did it because, the veterinarian said that. So there has never been an agreement of One Health in antibiotics. It is difficult at present. We are all working in One Health perspective. We started in Europe working in this type of ecosystem when we started working to fight against resistance. There are critical in human beings, but also resistance in the environment, in animals, and also in food. And of course, we started more or less in 2003, 2004, 2005. And as I said, this concept has been widely spread. So we know that there is a consistent flow between bacteria and antibiotic resistant genes, as we will see between humans. We have humans in hospitals, the animal community, companion animals. We're not going to talk today about this, but this is true. We could have also a transmission there with agriculture and production animals, and of course with the environment, and we're going to talk about this today. And as we can see, we have seen that this environment is paid more attention on today. The important thing is to treat a patient or animals, to produce animals. And we see that there are things that affect the environment that would be useful if we really approach them and know them, and could help us to cure the environment and animals, also in human beings, of course. That's why this is so important. So what is the role of the environment really in the resistance of antibiotic? We have already started in a One Health plan since 2014. And the example here is a little example with colistin, that probably is an antibiotic used in all the countries that are here with us. When we had a high level of, for example, antibiotics, when carbapenems, for example, when there was resistant to carbapenems, we started using colistin. Up to 2010 was used. It was too toxic, but since we didn't have any other antibiotic available, we started using colistin. Of course, it was used in animals, and also in those years we discovered a mechanism to resist to colistin. The result is that we have looked for these resistant genes to antibiotics that are horizontally transferred in the environment, in rivers, and also in wastewaters in the region of Barcelona, to know whether these antibiotic-resistant genes are in the environment, in rivers, in natural waters, for a biological function, could be, or if really they are associated to human beings. And for this purpose, we use isolates in rivers, as in wastewater in Barcelona, to see whether we had these colistin, mobile colistin resistant genes. And what we have seen what we saw is that we have zero bacteria resistant to colistin in rivers. That means that there is a direct relationship between human population and anthropogenic contamination, and also the presence of these genes. So these genes are a good marker of the use of colistin, because really we're using colistin in human beings and in animals. What we have is a high number of Escherichia coli intestine bacteria with these colistin resistant genes. We had Escherichia coli in the rivers, but none of them had these resistant genes to colistin. So there was a direct relationship between the contamination made by menin and resistance to colistin. The good news is that we have reduced the use of colistin in Spain, and we have also reduced these colistin resistant genes. So it is an excellent marker to know we reduce the level of colistin, we reduce these antibiotic resistant genes. The bad news is that this does not happen with all the genes and mechanisms of resistance to antibiotics. Or mechanisms are so happy, let's say, the bacteria with the bacteria. So even if we reduce the use of antibiotics, we are not going to reduce the resistance to these bacteria. So that's why I given you this example. This is a marker, it's a great marker for the reduction of the resistance to colistin that we have to do in other countries. I already talked about this, and we published this. We made the genomics of this, and as you can see here, we can see how we're capable. If we take this bacteria and see the level of antibiotic resistance, we learn a lot. But if we also sequence and know the genetic context, we learn much more. Here, for example, we see that in the rivers, enterobacteria, the main enterobacteria were Klebsiella in river, but also the bacteria that we have mainly in the bacteria of wastewater were Escherichia coli that have these resistant genes. All the bacteria of the rivers are sensitive to antibiotics? No, not at all. Here we have, for example, this gene RMTB in wastewaters in human beings, as you can see it, while this one was absent in river waters, although we have this gene here. With the same resistance to the same antibiotics. So also we may find not only resistance and susceptibility in different ecosystems, but also we may find antibiotic resistant genes different to the same antibiotics, that we can use them as a marker for the presence or absence of antibiotic resistant bacteria. We had, as we can see here, a population that was more diverse of bacteria in the rivers, and with fewer resistance or less plasmids to the bacteria that we had in our residual water, our wastewater plants to different types of E. coli. So we have a kind of bottleneck between the bacteria of the rivers and the bacteria that we have in our cities, because we only have those bacteria that are capable to colonize the intestine of human beings. Therefore, we have fewer clones, but we have more diversity in terms of plasmid and resistance to antibiotics. Some of the papers that we published in Venezuela, for example, we saw how we are capable to make a sample of one health, and we see how we find bacteria in a human being that are not really bacteria that are in animals or in the environment. These are bacteria that are specifically adapted to the colonization of human beings. However, when we make the sequencing, we see that they have plasmids of animals, as we can see here, this bacteria, E. coli, MCR1, it has this carbapenemase. So we can think, well, this is a human bacteria. It doesn't have any relationship to what is happening in animals in the environment. It's not the case. This bacteria has the same plasmid as this one in, for example, swines in Venezuela. So using colistin in swines in Venezuela, they have selected a plasmid, and this plasmid with this horizontal transparency of genes that are commented at the end ends up there active within Escherichia coli, specifically adapted to colonizing human beings. The last paper that we published in this sense is to see the importance of wastewaters are the hospital wastewater that are really very, very important. The last resource antibiotics The places where we use the most, obviously, is in hospitals. So we have made a paper in residual, or excuse me, in wastewaters of hospitals of Ghana, a rich country of Africa. And also we got wastewaters before the hospital, in the hospital, and after the hospital, and we see what bacteria can we find. And as we can see here, the hospitals really released wastewaters bacteria that are resistant to carbapenems. That means bacteria resistant to last resource antibiotics, and then they maintain downstream in wastewater. How do they maintain? How do these antibiotic resistant genes and bacteria resistant to antibiotics maintain in this wastewater? This is what we wanted to understand, and therefore we have looked at what bacteria we have. Well, we have basically two different cases that I want to show you to see the complexity of the system, but also that we have information that it is a must. We have high levels of resistance, new genes, but also we have two types of dissemination. One in pathogen bacteria. So one pathogen bacteria that comes out from the hospital, and this bacteria maintains, and we can find it in wastewaters. But in other instances, this doesn't happen. What happens is that we have environmental bacteria, not pathogen bacteria, environmental bacteria in these wastewaters have gotten these antibiotic resistant genes, and these genes are maintained in these cases. That means that when we are looking for bacteria resistant to antibiotics in our wastewaters, we should not rule out the bacteria that are used to living in the environment, because it is very possible that these are the authentic reservoir of antibiotic resistant genes of last resource. These are several examples. So that means that we have also a clonal expansion in wastewater of some bacteria like Pseudomonas, but also we have environmental bacteria that have never produced any disease, and that, however, are the reservoir of new antibiotic resistant genes that had never been seen before. So wastewaters and from hospitals are probably the hotspot, most important around the world in the generation of resistance to antibiotics. And to know this genetic information of each bacteria, we use hybrid sequencing, considering the bacteria. La Illumina is the that we use most of the times, and another technique of sequencing of long change plasmids, for example, and combining them, we may know what the genes are in what plasmids, what genes are in plasmids, and what genes are in the chromosome. And just to end my presentation, what has happened with COVID? COVID has increased the use of antibiotics, as we have seen here, not only here, but also in Spain. In March 2020 was our first month with COVID patients. We increased the use of colistin, erythromycin, and also last resource antibiotics. So in all the world, as we know, we have had more COVID, although the COVID is not treated with antibiotics, but we have learned during the COVID how to treat patients with antibiotics, because we had new viruses and we didn't know how to treat them. Then we learned that that in the pulmonary pathology we have to use cytokines, etc., to base the prescription. So it's very important to know this. When we have this population of bacteria in our hospitals, we have had more effectivity in hospitals and more use of antibiotics. In spite of all that, I think that the vigilance of antimicrobial resistance is very important. First, because we may know the early emergency of some bacteria. Also, it allows us to follow up, as with colistin, how effective are the measures to control the antibiotic resistant bacteria that we have in animals and in the environment in the country. And also, we can know that we are covering healthy population when we talk about antibiotic resistant microorganisms. We talk about hospitals, but we have to know also information about the healthy population and the tools that we may use. There are many people that tend to give lessons about how to make vigilance in the environment, and they say we have to use metagenomics, but I don't think so. I think that is not strictly necessary. We need a selective growth, a check and learn about the selective growth markers, pathogens, and also use molecular techniques, simple techniques, PCR, for example, mediated techniques to classify the bacteria, and if possible, the use of genomic and metagenomics, that would be helpful. And with this, hopefully I convinced you that there is a consistent flow of bacteria and genes and plasmids resistant to antibiotics in all these ecosystems, and that the real way to fight against them is to have a 1-8. This is the group that I lead in my university, and anything you need, you can contact me. And thank you very much for your attention.
Dr. Gonzalez, thank you so much for your excellent presentation. We have received a few questions from the attendees. I will read them now. The first one, what do you mean by level of resistance higher than 50%, the number of isolates in that species? Quantitative. The isolate resistant, we isolated 50, there are 50 bacteria which are resistant. This is what I mean. The number of isolates, percentage of the number of isolates, that's what I mean. Second question through the chat. Seeing the presence of MRCR1 irrespective of those which are resistant to colistin. No, we selected with resistance. No, we selected with colistin to see which bacteria had colistin present with a very good marker. Then in parallel and looking for other resistance mechanisms, we use other resistant genes like R1. So R1 does not have a high level of resistance. You need to find the adequate level of resistance to identify the bacteria of colistin. There are a few that have MCR1 and we can get lost. So this is a very good method that can identify that. The third question, I'm sorry, Dr. Gonzalez, right now we have 130 attendees through YouTube. You said that hospitals are a key element in the system. Do you believe that it would be important or interesting to treat wastewater when they leave all the way to the sewer systems? Yes, of course. The legislation makes it mandatory, but we know that it is legislation which is not enforced. We know that there are many hospitals in Spain that do not have the capacity. This is mandatory in Europe, but it is essential to do that. And there are many hospitals that don't have the capability of enforcing that. I will send you another question through the chat, but by virtue of the time, I would like now to give the floor to Dr. Zanoli so that she can also introduce the second panelist for this morning. Thank you so much, Gonzalez, again, for your excellent presentation. Thank you so much. Maria Inés, you have the floor now.
Joining the workshop now. Thank you, Julian and Marcos, for this important initiative to discuss waste-based epidemiology at the Latin American level. And also, I'm pleased to chair the coordination of the section with my colleague, Professor Aldo Cagero from the Laboratory of Environmental Biology of the Institute of Biomedical Sciences and of the Faculty of Medicine of the University of Chile. And now let's move on to our next speaker, Professor Lars Entrance, who will speak about genomic surveillance, complementary of environmental epidemiology in national public health systems. Professor Enstrand currently holds faculty positions at the Department of Medicine, Baylor College of Medicine, Houston, Texas, and New York University School of Medicine, New York, United States. He's the director of the Clinical Genomics Facility at Science Life Lab and director of the Center for Translational Microbiome Research at Science Life Lab at Karolinska Institute in Stockholm. Professor Lars has a large experience in clinical microbiology, and in a number of his studies has combined population-based epidemiology with clinical and basic microbiological science, including molecular biology and genomics. Dr. Lars, please. Dr. Lars, your microphone open.
Yes, can you hear me now?
Yes.
Okay, thank you so much. And I want to thank the organizers and especially Marcos and his team who invited us to be part of this Panakeam network. And I was asked to give some background how we responded to the pandemic in Sweden, describing the infrastructure we have, and also talk a little bit about the future, how we can use these capacities for environmental samples to track not only viruses, but also antibiotic resistance and more. So my background is MD. I'm a professor in clinical microbiology, and I have been in the microbiome field for almost 15 years. We started this Center for Translational Microbiome Research, and then my group is actually a research group from the beginning at the Karolinska Institute. The thing is that we have great experience with studies of large population-based clinical human cohorts. So the infrastructure has been designed for high throughput sequencing pipelines. But on top of that, we also have a culturomics pipeline for high throughput culture of bacteria and also viruses. I will give a background, and you all remember in the beginning of 2020, we were visiting a lab in China, and then we were looking for the infrastructure that we were interested to acquire to our lab. We had some of these robots and these sequencers, but the aim was also to start up a project called the Million Microbiome of Human Project. So the aim is to shotgun sequence metagenomics 1 million human microbiomes to get some kind of atlas about how is this microbiome designed in different ethnic groups and so forth. So this was the purpose with this meeting. And at the same time, when we were in Shenzhen, there was a rumor or rumors about the new virus that caused severe pneumonia in Wuhan. And we didn't think more about that, but you all know what happened in the beginning of 2020 when this virus was declared as a public health emergency. And also we had this pandemic in not only in China, but it was spread to Europe, and then we all know the story. So what we was asked to do, since we had access to high throughput capacity for analysis and sequencing of microbiome, we could see that we could use this pipeline also for COVID or SARS-CoV-2 diagnostics. And you all know that it's not rocket science to actually amplify, extract RNA, run qPCR and amplify the target, and then report back to the patient. So this was a quite straightforward thing. So we decided to look at what's in our infrastructure that could be used for these kind of diagnostic tests. And the aim was to reach 5,000 analyses per day. And at that time, the clinical microbiology lab could not deliver this amount. So there was a need for a high throughput capacity in Sweden. We didn't have all equipment at that time, so we were actually asked to ship more material and sequencers and qPCR machines and these robots from China. So we got a grant for 6 million euros from the Wallenberg Foundation in Sweden, and that helped a lot, and we could get the equipment to Sweden within a week thanks to this support from the foundation. And then everything started. We turned our research lab into a high throughput diagnostic lab in a couple of weeks. And we also set up this sequencing facility on the lower left. And you can see that we were not so sure about security at that time. So we were protected and had this protection gear around us. But the sequencing capacity was set up in parallel with the diagnostic capacity. So in 2-3 weeks, we had a couple of more units of these robots, because high throughput capacity requires that you have these robots. You can't do these tests or analysis by hand or pipetting. So we had this kind of approach to set up a high throughput lab in a very short time period. So in 4 weeks, we were actually up and running with a diagnostic lab in Stockholm, Sweden. And then we had to increase the number of colleagues and full-time employees, not only for testing and running the qPCRs, but also for packing of sampling kits and so forth. So we were increased from 35 people to 250 people in just a couple of weeks. And you can see we were quite busy. And this was during the second wave. The first wave was a little bit less samples because the regions in Sweden were not really prepared to use our capacity at that time. But you can see here that it was a busy schedule for many people in the lab at that time. We were quite aware that this could be a second wave, and this was actually also what happened in Stockholm when people started to go out dancing again. You can see the bars here that the number of positive cases in the end of or middle of the end of 2020 increased. So we had to adapt to that and adjust our capacity even more. So you can see that we diagnosed almost 600,000 plus samples at the end of 2020. And you can see the number of positive, 60,000 positive at that time. And it's clear also in the bottom that you can see the red bars that actually increased in the end of 2020. But at that time, the clinical microlabs were had a higher throughput than in the beginning. So we decided to, so to say, switch gears and focus on sequencing, because there were discussions about the importance to keep track on variants of concern. So at that time, there were not too much sequencing in the country, but I also argued in the newspapers that we need to increase this capacity, and we are ready to start up the high throughput sequencing pipeline and replace the diagnostic pipeline. So you can see here that we had a similar approach to get samples into our lab, but we skipped then the upper part with all the 600,000 diagnostic tests and focus on the sequencing part using our infrastructure. And at that time, we also have trained our technicians, and we have optimized the sequencing pipeline based on a special technology, and I will show that later on. Also, we had access to a very high throughput sequencing infrastructure. You can see some of the machines here, and the capacity is enormous if you compare to what could be done just 2 or 3 years ago. I mean, the T7 on the right can sequence 2,000 samples in 2 days, and that's what we are doing actually now per week. So there are a requirement to have this kind of high throughput capacity. And the good thing nowadays is that the sequencing cost is not that high anymore. So there will be lower prices for shotgun metagenomics in the future, and that will also gain what we are discussing here in this meeting when we look at environmental samples. So the setup for the COVID sequencing work is like this. We do not perform shotgun metagenomics. We focus on the target, that is the virus. And you can see in the yellow figure down here that we use a primer pool that capture the SARS-CoV-2 genome sequence. And then we have an artificial DNA and a human control in the same assay. And this allows us to use index for these primer pools, and we can run parallel samples up to 1001 flow cell. So we are actually now every week sequencing 2 flow cells on the T7. That means 2000 sequences per week. As all of you know, the mutations take place in the spike protein, the yellow part here on the right side. And it's in that part we find all the variants of concerns that have been reported, not only in Sweden, but also in other countries. And you can also see here that the dark dots here represent the mutations in the spike protein. And you can see the alpha, delta, and Omicron variants, that the Omicron actually mutated a lot in this region. And this is probably also the reason why it escapes the vaccination now, even if it gives a lighter infection and not that severe infection anymore. But to use this kind of approach, we can actually see if new variants are taking over. And this is also shown here. Well, what we had to do then was to use the high throughput sequencing approach. And we also were depending on a server that could handle all the data that was produced from this. And then we upload everything to the Public Health Agency for bioinformatic analysis. And of course, all this requires a very trimmed, so to say, pipeline with dedicated persons on all positions. And this is what we were setting up here, a prep team for reception and activation and everything that could actually prepare for 20 plates per week. That's approximately 2,000 samples. And then they move that into the sequencing team that has an extraction, and then the sequencing where it takes place. And then after this, we go over to the data delivery team, and everything is automated nowadays. So the sequencing generated on the T7 sequencers are automated, uploaded to the Cytron server, and then to the Public Health Agency. So this takes from start to the end approximately 1 week, and this is because we need to optimize and use the flow cells in a very optimized way, so to say. And the interesting thing is that we get access to almost entire Sweden with this sequencing platform. We have 21 regions in Sweden, and we cover and get samples from 19 of these. And as I mentioned, we have approximately 10% of all positive samples from each region based on the population size. So this gives us 2,000 samples per week, but theoretically we can go up to 10,000 sequences per week if required. And this just show how the spread of the virus and the different variants takes over. And this movie shows from the beginning, we had all these gray sequenced strains that actually didn't have a name. And this happened up to November 2020. And then you can see that the new colored viruses takes over. And then we have the beta and the delta version coming here in the 2021. And then we sequenced a lot of the delta versions, the blue ones. And now in the end of 2021, the Omicron actually takes over. So this clearly shows how dynamic the process is, and the importance to track the variants of concerns. And this is what we are doing now on a daily basis here. And you can see here on this screen also that the alpha variant actually disappeared, and then we have the delta variant took over in October 2020. And then it was dramatically a difference here with the Omicron at the end of 2021. And now we can see that Omicron is totally dominating in Sweden today. And we can also see that the vaccine was designed this period. And the third shot for most people were in this period. And still the Omicron can infect and spread very efficient. So this is a situation that we have in Sweden, and I'm quite sure the similar situations are appearing in other countries. This was a couple of weeks back. We sequenced 100,000 COVID whole genome sequenced viruses, and today I think we have 120,000 sequenced. This was very. These are the number of sequences per country, and you can see that Sweden has a very high position now, mainly thanks to what we are doing here, since we are sequencing more than 70% of all positive strains in Sweden in my lab. So what is happening now? Well, we have named ourselves the National Pandemic Center, and you can see on the top that we are still sequencing the virus. We are also establishing a COVID-19 biobank, and I will come back to that. Then we will be now also a preparedness center for diagnostics, since there may be a new wave after the summer. Now we don't see that high numbers anymore, but we will be prepared here. And then, of course, we want to have a research track in our lab as well. So if we look at the biobank, we sampled all and stored all these 620,000 samples, among them 60,000 positive. And then last year and this year, we have approximately 550,000 positive samples, and 110,000 of these have been whole genome sequenced. And this will be also in collaboration with Biobank Sweden and regions, and the samples will be accessible for researchers to study not only the sequencing of the virus, but also, for example, if the microbiome itself in the throat could have an impact on the severity of this disease, for example. So there are lots of opportunities here with these stored samples. And when I come over now to surveillance and early warning of COVID-19 in wastewater in Sweden and abroad, this was a European Commission recommendation in the beginning of March 2021 to put in place a surveillance system and then cover a sufficient part of the population and the numbers of samples per week to keep this kind of control, and then use common methods for sampling and analysis. And of course, now when we have this network, Panacea in South America, this could be applied also for us in this network for sure. And this is how we see it. I mean, it's not, as we heard, only surveillance of the virus. It could be other pathogens, and especially resistant gene surveillance. I believe we can set up a quite good system to catch the genes of interest, the resistome, and maybe avoid shotgun metagenomics. That is a little bit overkill, but if we focus on the resistant genes that we are after, we can set up a similar autoplex, multiplex, PCR-based sequencing approach to catch the resistant genes in that environmental sample. And this is, of course, something that we are setting up now when we have some test samples from this network. And I believe that sample collection is extremely important. We have to streamline this and also have a common rules how to sample and how to prepare the DNA. But then we can also do that on site, so to say, and send it over to us for high throughput autoplex library preparation, and then do the sequencing in-house, so to say. So this could be a resource for the environmental wastewater-based epidemiology covering many countries. And you can see here also the benefit with this multiplex PCR-based sequencing approach. We can have many, many more targets. For this traditional way of identifying targets like 2 resistant genes, here we can come up to 400 resistant genes. And the sensitivity is much higher with this approach. And you can also see the time is a little bit longer, but if this is a research project, it's of course not so depending on rapid turnaround time. But I think this is very important, the genome assemble possibility with the multiplex-based sequencing approach. We can also study then where these genes are located in a certain species. And this is something that can add to the assays that are commonly used today. And this is another approach that we can follow, for example, Omicron or other variants. from people arriving at the airports. So we take samples from the wastewater and have a similar approach here. So this could be a way to control for new variants coming into the city or the country, and it could also be a way to study the transfer of resistant genes from different areas in a quite convenient way. So the same approach here with this autoplex amplicons, and we are setting up this now, and we hope that we can have it up and running later this year. So I just want to finish thanking the pandemic team here, and these are the core people working with me, Jessica, Mike, Marika, and Frederick. And we have lots of collaborations here, and this is the National Pandemic Center, and then we try to combine this with the microbiome infrastructure. So this is the whole group here in Stockholm, and we are now focusing not only on the human microbiome, but also on pandemic preparedness. So thank you so much, and I can take some questions.
Thank you, Professor Lars. Really, it was a pleasure to hear you and listen your deep experience in genomic surveillance. Really, this is a very powerful tool in the epidemiology of all the diseases from pathogens and antimicrobial resistance. Thank you so much. We have here 3 questions that came from the chat. The first one, I think that you have already said something, is how the MGI technology works in comparison to more conventional like Illumina or PacBio, or the sequence results different, the same pipelines works?
Yes. I mean, the amazing thing with the MGI platform is that we have a much higher sensitivity for virus sequencing. I mean, the other platforms cannot take with a high quality CT values over 30. We can sequence whole genomes with a 98% success rate up to CT 35. So this means we have a higher quality and a higher sensitivity with the MGI platform. And we also benchmarked with other platforms, and the MGI is cheapest and superior in our hands, at least.
Okay, thank you. The other one is the drop in sequence will allow using deep sequence for environmental surveillance. This is the answer that I would like to hear. And how long do you need to train a new generation of data scientists in environmental epidemiology?
Well, the last question is a big bottleneck for all of us to find scientists who actually can handle this, bioinformaticians. This is we lack that kind of experience also in Sweden. But I'm quite concerned that there will be pipelines for analyzing these kind of samples in the near future. So you don't have to be a fully trained bioinformatician to actually interpret the data and make it possible to study these samples. But at the same time, of course, cost is another drawback. But the cost is also decreasing all over the time. I mean, we sequence 20 million reads for approximately 40 euros, and that is a tenth of the price to a year ago. So prices are coming down, and the sequencing depth is, of course, something you need to standardize for environmental samples. But for a human microbiome, a fecal sample, we can get 98% microbial DNA and sequences, and then that's quite a good coverage with 20-25 million reads. So that is the cutoff for human samples. But for environmental samples, I'm quite sure we can set up a similar thing. But for the multiplex PCR approach, you don't have to sequence that deep to catch what you are after. If you know what you are looking for, you can actually have 400 or 500 different targets and then sequence for a couple of euros. So it's really a high capacity that is in front of us from now.
Okay. And the other one is related to this one. And can it be cheaper and easier for Latin America countries to use waste wastewater-based epidemiology instead of clinical trials?
If it will be cheaper,
Yeah, cheaper and easier.
yeah, I'm quite convinced that this technology has been now available for a couple of years, and the price and the throughput and everything is improving. So what we just could dream of 3 years ago is now reality. So I'm quite convinced that this technology can be transferred, and we are very happy to provide knowledge transfer of what we are doing here in this network. So that's what we are doing currently now together with you.
And the other one is, what's the main sequencing platform you use to assemble SARS-CoV-2 genomes, and why?
The bioinformatics.
The sequence platform, yeah.
Yeah, I mean, it depends. If we look at the metagenome pipeline, I'm not an expert exactly what kind of, it's improving all the time, but that is available on our web page if you wanted to look. For single isolates, we use the traditional pipelines that have been out there. But for metagenomic analysis, these kind of pipelines are improving all the time, and there are lots of ads that you can look for. But we have all this on our webpage if you're interested, and they are also mostly free available on the web. So it's not a big problem to get access to the pipelines.
And I have I do the last question because of the time. To carry out a metagenomic virus analysis of a wastewater sample, it is necessary to carry out a previous enrichment of the samples?
If you are talking about bacteria, for example, no, I don't think so. I mean, we don't do that here. The way we actually target is to use the multiplex PCR approach, and then we, so to say, amplify the genome of that virus. So this approach can be, now it has been designed for SARS-CoV-2, but we can do it for influenza or other coronaviruses as well. As long as you have the sequence of that target, you can perform this kind of multiplex PCR approach on whatever you look for. Another approach is to go extremely deep with the metagenomic sequencing, and then of course you will find the virus, but we benchmark the autoplex with the shotgun sequencing, and I mean the sequencing depth is enormous if you should find very low amounts of virus. So we believe the autoplex is superior to other approaches here. But we will see. We get samples from South America now, and of course in the wastewater there are lots of very few viruses. And the normal way to do this is to run qPCR, but we hope that we can add sequencing information on variants also, especially new variants, because the mutation PCR is just giving us what we know is out there now. But if we want to see new variants, we need to have a whole genome sequencing of the virus for sure.
Yeah, I have more questions here, but we'll try answer after. I can send to you, and perhaps you can answer direct for the person. But I would like to share one comment here. Lars' amazing work and lovely story from before the pandemic to the future of the pandemic plan. What a journey from Doug Manuel. I think it's really very, very exciting to see all your history since the beginning of the pandemic till now. And thank you for sharing this with us. Thank you, Lars. So thank you.
And good luck with the meeting. I will try to be part of it, but I cannot make it the whole time. But thank you again for inviting me, and we are very happy to be part of this network. Thank you.
Okay, thank you.
Muchas gracias, Lars. Muchas gracias, Bruno. Gracias, María Inés y Aldo.
Thank you, Lars, Bruno, María Inés, for this session, for being the moderator. We have a 5-minute break to prepare the next speaker. We still have many questions in the chat box, but as part of the network, we're going to make a brief summary or a briefing of what this workshop was, and then we will try to respond most of these questions when ready, and we will deliver them to you and to all the attendees, to all the people that registered. Over 400 people registered and 150 live in YouTube. Thank you very much again. And we have a 5 minutes break for coffee. I'll see you in a moment. Thank you. Thank you. Thank you. you to the session 2 of the workshop. My name is Veronica Rajal, and I have the honor of being the moderator of this session together with Dr. Pablo Sotelo. Dr. Pablo Sotelo is a biochemist in science, a PhD in microbiology, a professor and head of the Department of Biotechnology in the Science Chemical Department of Asunción, Paraguay University. Also, in CONACYT, Paraguay, he is a member. So the word is yours, Pablo. Thank you very much, Veronica. A pleasure to share with you. Veronica Rajales, engineering PhD, the National University of Salta, investigator of CONICET in the research investigation in Salta, Argentina. We are moderators of these talks that are going to be in the second session of the Panacea program. We have now 3 presentations coming. Before we start with the first one, I want to remind you that you can log in. If you are logged in the YouTube, you can make questions through the chat that are going to be answered at the end of the presentations. I am gladly very happy to introduce you to Tatiana Prado. We have known Tatiana from the SARS-CoV-2 wastewater group investigator in the Laboratory of Comparative and Environmental Virology, Osvaldo Cruz in Rio de Janeiro, Brazil, experience in environmental microbiology, environmental health, worked with vigilance in the environment and also virology. And SARS-CoV-2 in the wastewater has collaborated with the OPS, making a guideline about SARS-CoV-2 in wastewater for Latin America and public actions for the control of COVID-19. She's going to be talking about the environmental surveillance of pathogens in wastewater in support of public health with emphasis on SARS-CoV-2. Tatiana, we're happy to have you with us. Thank you very much for participating in this workshop, and we are here to listen to you. I would like to thank you for giving me the opportunity to participate in this workshop that is going to cover a very important topic. Currently, my presentation is going to show what we have learned from the vigilance of SARS-CoV-2 in wastewater, and also data reported in the latest year, and also the models presented here can expand with certain level of customization to other pathogens that may be present in this matrix also. Analysis of wastewater, looking for evidence of pathogens. We have a long history of using public health, but the most well-known example was the use of this approach in the initiative of a worldwide eradication of polio. However, it was with the pandemic COVID-19 that this approach of environmental vigilance was more important, with many experiences reported by many countries with interface of the public health policies. That is why we concentrated in better understanding these experiences using SARS-CoV-2 as a model to widen this type of vigilance to other pathogens that have ways of transmission that are oral or can be extracted by urine or feces of infected tissues. The scientists of all the world are coordinated efforts to gather data and foster collaboration amongst all the people involved in this field of investigation. COVID-19 with water-based epidemiology has been launched with the purpose of potentiate the collaboration at a global scale for epidemiology of SARS-CoV-2 in wastewater. The group now includes more than 500 collaborators from all the world, including some of the collaborators here. Here we have, for example, COVID-19 with the locations of 64 countries that analyze their wastewaters in more than 276 universities and more than 3,300 sites in all the world. This figure represents the coverage of the population using different approaches of vigilance of COVID-19. Well, the population treated by hospital vigilance, clinical tests, also sewage vigilance of wastewater, of course. Also, these charts represent the blue one, the population currently assessed by vigilance, And through this figure, we can see that all the people treated by the sewage system can be assessed for COVID-19. So with this approach, it's possible to assess people that are asymptomatic or that do not have access to clinical tests. Some benefits of the wastewater tests include a wider detection, lower costs is very important. A recent World Bank report showed that per person per year making the sewage test is approximately point or half of a dollar, in the clinical test could be $5 per person. Also, the control of the pandemic, early detection allows the control of the preparation of the healthcare system through the identification, fast identification of routes of outbreaks and waves, and also could be more democratic because the test of wastewater include all the people and have the capability to be targeted to vulnerable populations. Well, based on the experiences that we have gathered, we have developed a flow chart to exemplify what would be a wastewater vigilance program and also public health actions to control outbreaks or epidemics. Some of the principal elements would be the sampling, also laboratory controls, also the interpretation of results, information conveyed to different groups through institutions for the decision-making process, also leading to some actions of public health, amongst them active vigilance, social distance, lockdowns, risk alerts, also assigning resources to the development of strategic areas like, for example, health care services. The OPS elaborated guidelines or reports supporting the vigilance of SARS-CoV-2 and other pathogens in wastewaters, with the support of many investigators, mainly in Latin America. The first technical guideline is focused on the analysis and quantification of SARS-CoV-2 in wastewaters, and including, for example, gathering the sample or collecting the sample, viral concentration methods, ribonucleic acid purification, and PCRs, inverse retrotranscription PCR controlling and assurance, quality assurance, and biosecurity control. The second guideline is focused on a basic problem of vigilance of waters and also public health action with focus on Latin American countries, and includes the following topics: introduction, applications of vigilance based on wastewaters, SARS-CoV-2 vigilance in wastewaters in Latin American countries, implementation of the environmental vigilance programs of SARS-CoV-2 and other pathogens of interest for Latin America, coordination of the vigilance program based on wastewaters, a management system of information about environmental health, communication of vigilance data to inform and support actions of prevention and disease control, profitability of the program of vigilance for SARS-CoV-2 and other pathogens of interest, ethical and legal considerations and topics under development. The report starts with an introduction and applications of the vigilance of wastewaters for the healthcare system, and also with three important topics: early alert and preparation of the healthcare system, with also temporary monitoring and space monitoring, and also evaluation of new pathogens or re-emerging pathogens to prevent new outbreaks or pandemics. Also, we have the size of the population in the site of the sampling lead to different actions of public health. In terms of public health, in populations connected to a sanitary sewage system, the experiences have let us know that we can make treatment plans for wastewaters, also sewage piping in close places, like, for example, buildings and installation in general. The the waters that are released have a high number of affection in people, more than 50,000 people, while in the piping, a higher number of people, usually less than 10,000 people are affected. Some examples of literature have demonstrated that for the control of the dissemination of the virus in specific areas and focalized actions of public health, the analysis of a pipeline and sewage with a fewer number of people would be better if in terms of the sampling for this purpose. But we have also to take into account that an important part of the population of Latin America is not covered by healthcare services, mainly sanitary sewage. This graph represents the evolution and the percentages of homes with access to basic services. The blue line represents the percentage of the population covered by water services, the red line by electricity services, and the green line by healthcare services. It's possible to see that about 40% of the population is not covered by healthcare services or appropriate healthcare services. In this sense, we could see some experiences of SARS-CoV-2 vigilance in also sewages of vulnerable populations or rivers or creeks that are not treated in terms of, not treating the water. The first case is an example of wastewaters in Quito, in Ecuador. These are wastewaters that are not treated, a recollection in 3 different points of the river during the period of June 2020, when we had more than 4,000 cases of COVID-19, and they detected high concentrations, viral concentrations in the samples that reflected the circulation of the virus in the area that was sampled. A second example was also in the city of Sao Paulo in Brazil. The samples that were collected, or an example was the sampling of a creek of wastewaters in a community that is a vulnerable community, 8,000 people in the community of Brazil. The samples were collected from May to November 2020. And in this graph, we can see the number of cases of COVID-19 in blue, and the green lines and orange lines are the concentrations of SARS-CoV-2 during a period of the monitoring period. The authors concluded that the viral concentration of the samples followed the epidemic curve of many areas, confirming that the vigilance can be made even in places that are not covered by sewage networks. In several countries of Latin America, we could see different scenarios in terms of epidemiology, including experiments of wastewater vigilance experiences and experiments, also vulnerable communities, for example, reused water, irrigation. We have important examples provided by Mexico, for example, recreation waters also, and we have other enteric virus and other pathogens in subterranean waters. Since 29 experience gathered in some countries that provided data or had published studies like Argentina, Brazil, Chile, Costa Rica, Ecuador, Mexico, Peru, Paraguay, and Uruguay. We summarized some of the parameters that were used during the vigilance of wastewaters. Most of the experiences were developed in urban centers with the wastewater areas, treatment plants with different populations from 2,000 and up to 10 million inhabitants. Also, when the collection of the samples were done in sewages, the populations included a fewer number of people, like you can see here in the chart. In rural areas, also in the population is less, it's fewer, as we can see here. Also, we recollected a volume of 1 liter volume of wastewater, the same vary from 250 and 2 liters in most of the trials. And most of the experiments were performed with composite samples. The frequency of the sampling usually was weekly or every 2 weeks. The concentration protocols in years of detection were based on the OPS, and the duration of the monitoring was between 6 months and 1.5 years. We have proposed a model for the vigilance of wastewaters in different stages of a pandemic. In the initial phase, the objective would be the early alert. In the acceleration and deceleration phases, it would be assess tendencies, and a preparation phase for future pandemics, assessing the reappearance or reintroduction of a pathogen. Then we talked about the target population, sentinel size, where to gather the samples, frequency, and method for this sampling process. Most of the countries have assessed temporary trends in terms of territory involving sewage information in treatment plant of wastewaters with a population that is usually more than 10,000 inhabitants in urban centers. In addition to the experiences that we have reported, we have several trials that have been published or presented for publication that contain this topic in Latin America. Most of them show the correlation between the data of viral concentration in wastewaters and the number of cases of COVID-19 in the communities, demonstrating that vigilance of wastewaters is a very useful epidemiological tool. Most of the experiences involved public resources financed by the states. However, some countries created alliances with other countries or international agencies for technical scientific assistance and also financing assistance in supporting monitoring. Most of the experiences were studies performed or conducted in universities or public research institutions, and the monitoring vigilance monitoring data are shared with the public health system, but the data in most of the cases have not been supported by direct actions to control the spread of the virus. One example of wastewater control and monitoring for public health against the propagation of the virus in vulnerable communities is Niterói in Rio de Janeiro, Brazil. During the first phase of the pandemic, monitored the quality of the wastewaters where they didn't have any case of COVID-19 notified yet. In this point, it is very important to highlight the participation of the healthcare sector that contributed not only to the follow-up of the areas of interest, but also the activation of all the healthcare network. After the vigilance of sewage systems, they found a high concentration of virus in this community population, average population of 2,000 inhabitants, allowed activate the vigilance system through the family doctors, a program established in a healthcare system in Brazil, to perform an active search of cases in the region. Some suspicious cases are patients that presented, for example, typical symptoms of respiratory acute symptom were transferred to healthcare systems to be assessed. And they have also, contacted and monitored these patients looking for primary health services for the follow-up. For patients that were positive, they offered also lodging in hotels in low-income families that could not make the social distance or social isolation process because the families living under conditions of extreme poverty, and the prevention measures and basic healthcare services are aimed at reducing the number of cases. And the next weeks, also this reduction was seen with the drop of the concentrations of SARS-CoV-2 in the wastewaters produced in this community. And as you can see in this graph, after 3 weeks of monitoring the concentration of ribonucleic acid in wastewaters and also the use for this community demonstrated that the preventive measures were effective to prevent the spread of the virus in the community. According to the experiences that have been reported, genomic vigilance or sequencing for the evaluation of variants in wastewaters presents some challenges for most of the countries, which are, for example, methodological problems to provide a response in real time, the lack of laboratory structure that implies the acquisition and maintenance of costly equipment, cost of acquisition of inputs and reagents, and the investment for the contracting or hiring and training of human resources and financial resources. Now, the crisis of public health systems associated to the crisis of the pandemic has created several important problems to track the progression of COVID-19 in the communities. In terms of the wide diffusion in electronic media or in communication means, most of the experiences involved reported the diffusion of the results of the areas or websites of regulatory, environmental regulatory agencies with the presentation of weekly or monthly reports about the dissemination of SARS-CoV-2 in the areas that were monitored. We have here some examples of, for example, alert bulletins to monitor the situation. Many municipalities are publishing the data together with other healthcare indicators in a kind of a dashboard fashion, showing the website, the results of the monitoring per region. Generally, they use a georeference system, and the data are translated in heat or risk maps present in areas with higher concentration of the presence of the virus in each location. The COVID-19 studies, the geographic information systems could be valuable tools in the decision-making process. And what is most important in the mobilization, social mobilization for the responses of the community to understand the dynamic of COVID-19 is essential for the mitigation of the virus. That's why these works that are performed in all the regions are very important and are performed in all the world. Well, now we have to have a close coordination between laboratories of investigation, whether public or private, the healthcare companies, public health institutions, and other institutions to guarantee that the strategies of the sampling are based on the public health needs, and the results are integrated with all the programs and are also linked to populations that are support. In spite of the technological advances, there are still some barriers for vigilance of wastewater to inform public health decisions. Most of the times, the information sources are out of the healthcare sector. This includes routine information gathered by different governmental agencies, universities, and organizations of investigation, private sector, and also service providers. It would be necessary to gather data at a local level and then join them to a national information system to group data and facilitate the following of the epidemiological situation, helping different institutional players in the decision-making process. Topics under development, there is a quantitative relationship based on the concentrations of the virus present in wastewaters. The estimation of an affected population. For this, we would need to determine the quantity of genetic material present in the excretion of people, and from the concentration found in the samples to extrapolate to the population using mathematical projections and trend curves. However, it's still difficult. There are uncertainties related to the variance and with the applications of the mathematical models that have to be developed throughout time. And we have here some examples in this slide. Now, concluding, some future uses of the vigilance of wastewaters include monitoring emerging diseases or endemic diseases, drugs, toxins, pesticides, also antimicrobial resistance that is very important, mainly also in this pandemic scenario. And also we have to consider an integrated one health approach, including the three types of vigilance: human, environmental, and animal, to build alert systems that are more efficient for future outbreaks or pandemics at a global level. Well, these were the main messages that I wanted to convey. Thank you very much. Thank you, Tatiana. We have some questions in the chat. The first one is if you have these guidelines available, these OPS guidelines, and where they can find them. If you could please give us the link, I can show the link in the YouTube screen. Well, the first guideline, the technical guideline, is already available. Well, Veronica, do you know if this is online? Can we share it? I really don't know if this document is online, but we have it. We have this document.
Está online todavía, la verdad.
Por ahí
Well, we can, I don't know if it is online, we can check that out. Maybe the people who required it, please send us an email and we refer it to you for you to send the guidelines to them if they are not available yet. The second report is in the review phase by experts and investigators. So in brief, we expect to have them published, to have these reports published. Well, I have here another question that was made. What could be the factors that really hamper the data obtained in wastewater in public health? The factors, excuse me? Yes, which could be the factors that really hinder the use of the data obtained in wastewaters in public health? Well, the lack of understanding, clear understanding by the healthcare sector of how to interpret the outcomes in terms of quantification or the meaning of risk, increase of the concentrations, which are the concentrations that determine an increase or an increase in an area, for example. An abrupt increase, these parameters need to be better worked and understood with the investigators in the healthcare sector. In the same sense and about the same topic, Tatiana, I think that is very important what you commented about the transference of the data and the interpretation. Now that you are commenting about the decision makers in public health, you showed several scenarios in the region, which could be the best experience of the best mechanisms to generate these kind of bridges to provide the information useful for or to inform the decision-making process. I think that, also there are risk maps per area, geographical region. This would be a very efficient way to communicate the information for the population in a general way, as this has been done by many municipalities and cities in all the world. I think that this would be the best way within the healthcare center. We're thinking about Rio de Janeiro. How can we include the data or why we have an information system for the increase of cases per region with the clinical tests. But we are thinking how we can or how we can do this, or with the results of environmental vigilance and wastewater vigilance results. how we can really use this and provide it to the public in with monitoring in determined areas in real time and with the data of environmental the information that we are identifying. We have one last question, and then we will move to the next presentation because we are just on time. But are there criteria to assess at the time of deciding the quality of the samples of wastewaters? Yes. Yes, the question is there any criterion to assess the time to decide the quality of the sample of wastewaters, a quality criteria to quantify it or analyze it? Yes, of course, even there is an obvious guideline that I mentioned with the participation of several Latin American specialists for the elaboration, experts in the area who participated, and some of them are present in this meeting. And yes, there are several tests for quality control, for example, those could be internal controls using virus that are similar to the virus of interest in the case of different specific virus. There are some examples in the guideline. They provide several examples, even for concentration, quality criteria. Everything is included in the guideline, and including several criteria for the quality control of these waters, including internal controls, endogenous controls, and also external controls. We do have a series of examples in this guideline. Well, we have an additional question, but since we are over our time, I'm going to read it anyhow, but I think that it would be very good to answer this question through another means later on. I'm going to read it. It says, about the methodological problems, do you think that it's possible to move forward as a region, also analyzing the stages and sampling? And well, I think that you have commented something about this in your presentation, but afterwards we're going to invite you to answer the question probably personally, okay? In order to continue moving forward with the presentations, if you agree, Pablo.
Great. I think that we can move forward, and this is something that we have already discussed in the guidelines of the document that was generated. So let's go to the next speaker. Thank you so much, Tatiana. It has been a pleasure to have you as a speaker. You have done a good work of integrating the different elements for the region. Thank you so much, Tatiana. Now I would like to go to the next presentation, which is the perspective of wastewater-based epidemiology, the World Bank Initiative. In this case, we have two different guest speakers. Doug Manuel, he's a distinguished professor of Ottawa University. He's a specialist in public health with 30 years of experience in development and assessment of surveillance systems in public health. He's leader of the wastewater group of the National Network in Canada for the investigation of variants. He's a consultant to the World Bank in Latin America and the Caribbean, co-leader of the Open Data for Environmental Surveillance in Public Health program used in more than 27 countries. We also have a second speaker for this same topic, who is, I don't know if my pronunciation is right. Perhaps it will be wrong, and I would like to apologize. John Martin Broyle, who is a senior water and sanitation specialist for the Latin American and the Caribbean region of the global practice of the World Bank for Water. He has more than 15 years of experience in leading and supporting loans, analytical services, advisory and consultation in engineering in Latin America and the Caribbean, Western and Northern Africa, and the Middle East. He has a PhD in wastewater engineering in Canada, and now he's seeing the treatment and reuse of wastewater, treatment of fecal and sanitation in countries, the intelligent management innovation of waters. I would like to give the floor now to start the presentation. Thank you so much. And in order to clarify, I'm going to make the presentation, and all this information will be available. And my colleague will be available to answer any questions and to share his experiences in Canada. I'll be doing this in English, but I may answer in both languages, English or Spanish, all your questions.
Thank you very much for inviting me to this very interesting seminar. I think I want to start by perhaps presenting a case that is not unusual, meaning that it is something that we've seen in other countries, but this is one that really helped us, at least within the World Bank group, to convince our management to embark on going a little further with financing wastewater-based epidemiology. And this is the case of the province of Ontario in Canada at the end of January 2022. While on the left side, you can see that the cases, the hospitalizations were kept on increasing towards the end of January 2022. But at the same time, the wastewater signal was indicating that the Omicron wave was decreasing.
John Martin, sorry, we are not seeing your slides. If you can share your screen, please.
¿Ahora la pueden ver?
Ahora sí.
Yeah.
Perdón.
Muchas gracias.
Okay. And then, so as I was saying, I think that the case that really, started sparking interest, at least within the World Bank's sort of higher levels of management, meaning that this could lead to an increase in interest in financing for wastewater-based epidemiology, was the case of the province of Ontario. Again, just quickly, you can see on the left graph that the hospitalization cases were still increasing in end of January 2022, but the wastewater signal had started to indicate that the Omicron wave was coming to an end. And the government of the province decided to use the wastewater signal to start taking decisions on how to reopen the economy and using that rather than basing themselves on the clinical cases, the hospitalizations, etc. And to us, this is a very powerful example of where, all, let's say, wastewater-based epidemiology pilots or programs should go. It's not to really inform the ministries of health and the economies on how to react and act and prepare. This is not a slide that is also unknown to most of us here. We realize that still more than 85% of the sites that are doing wastewater-based epidemiology are in upper middle income and higher income countries. And this is why also we got interested, because we wanted to make sure that knowledge and that experience is brought or shared with countries in other places where we work, where we concentrate our work. And so we started with 4 interventions, 3 pilots, one in Ecuador around, let's say the discussion started around April, May 2020, another one around the same time in Uruguay, and then also provided some support and financing in Buenos Aires to the AISA water and wastewater utility. And then we also did a small pilot in Morocco as well. And today I think what I want to do is to share some of these lessons and show how these sort of helped us design the next steps and our initiative, where we want to bring support to countries that want to embark on these approaches and particularly to scale them up. And some early lessons, and I think a lot of you are facing this or have faced this. So that we all know that this is a new ecosystem, and we saw this very acutely, let's say, in Uruguay and Ecuador, and we saw that there's a really important, that's very important to engage early with all the actors that are involved. No, and we saw that most of the initiatives were being started either by academia or by the water and the wastewater sector. And it was difficult after that to bring in the health authorities. And you really need a health champion to be able to take this to the next level. And here I also mentioned ownership of local actors. I think our approach at the beginning was to partner with external entities. We partnered in Uruguay and Ecuador with Biobot, and in Morocco with the KWR from the Netherlands. And we wanted to transfer the knowledge that they had and sort of validate the protocols that either they had started developing or help them develop their protocol. And I think the challenges here that we saw early and that we still, I think, face in many countries is that the health authorities are overwhelmed with the crisis. And it's very difficult if you don't engage with them early to convince them of the benefits of embarking on wastewater-based epidemiology. And so the communication is not only important for the population, communicating what are the results of these analyses and what they mean, but also we need to communicate these results effectively to the overwhelmed officials, and that's where most of our issues were when we did these pilots. And then the other lesson was, if we want this to continue, if we want this to be sustainable, we need to think about how to go from pilots to scale, no? And in our case, how to take these pilots and bring them into the broader projects that we finance in these countries. And so, for example, in Uruguay, we didn't have an operation, and we did a pilot with financing from us, from the IDB as well as from let's say from other partners. The CAF is interested in continuing this. So there was very much a very broad effort with lots of stakeholders, at least for the financing part, no? But none of us had a continuing operation that could help scale it up. And I believe it's still at a level which needs dialogue with governments to make sure that interest is sparked and that it can be brought to a national level and led by the Ministry of Health in the case of Uruguay. No? In the case of Ecuador, we were lucky to have an existing project, and so we continued, and it's been a year and a half now of monitoring in Guayaquil, the city that was the hardest struck, let's say, by the COVID-19 pandemic at the beginning, at least. And we have discussions with not only the health authorities, but the emergency committees that manage crises in a multi-sectoral way. And so we found a way to scale up, at least in Guayaquil. No? And another interesting case, here we didn't bring any technical assistance. It was really just financing towards the end, or let's say the middle of their experience. And I think it's an interesting point here, and I'll come back to why this is important later. My point here is we need to make sure that all the stakeholders understand the benefits and find something for them. No, let's say the phrase "what's in it for me" is really important here. And I found this example very interesting in Buenos Aires, that the water and sanitation utility, AISA, had already looked at using PCR equipment or let's say the broader molecular biology tools to enhance their own operation. So it wasn't, at the beginning, it wasn't necessarily to just apply it to the COVID-19 crisis, but they as part of their strategy, innovation and technological development was there as part of their priorities. And so they were able, because of that, to really quickly convince their management and to embark and mobilize the personnel to initiate these tests. And this shows the importance of thinking through also why are we doing this, what are the benefits that come with this, what are the benefits for me, and I think and ISA even went further. Now they're including wastewater-based epidemiology as part of their overall risk management strategy, which I think is an example to think about when we have discussions with water and wastewater utilities, who many times ask me, why should I do this? Am I just a sampler? Am I there to interpret the data as well? But is there something else? No? And then so we, with this experience, and then bringing in the experience from Canada through Doug, we published a guide, on how to take the available information that's out there and think through what could be a strategy to scale this up to a national level. And so here's a simplified illustration of what are these steps that a country can take to implement a wastewater surveillance strategy. And these are simple steps, but they're not easy to implement because of the multi-sectoral nature of wastewater-based epidemiology. And the first step is really just to assess what you have. What do we currently know? What don't we know? Who are the actors involved? Most of the time there are many. Can they be brought to work together? Should we avoid replication, or not replication, but doubling, let's say, the redundance in the responsibilities and what different actors are doing, and then really define what the priorities should be for this scale up. And so should it focus on the broad population, meaning as Tatiana was mentioning, how do we include the non-sewered population? What level of aggregation or disaggregation do we want this to help, and then start really engaging with all the stakeholders to refine and optimize the strategy. What do we want to monitor? What are the targets? What is the local capacity? And in some countries, for example, such as in the US, they brought in a private partner to help set up the news network at the national level, and then evaluate the strategy, adapt it, and redo the cycle. So it's very much an iterative process. And this is where I think we're not only interested, but I think this is where we can help in two ways. One is really our traditional role of mobilizing finance and knowledge, so bringing in capacity building, such as we did in Ecuador and Uruguay, building the right environment, which means bringing in all of the different actors along the decision-making chain to make sure that it is possible to indeed to embark on a scale-up process, and then making resources available, not just financial, but technical as well. For example, here you see on the bottom left side the report that we published recently, really for a Latin America audience, but this is also available for a global one. I think the lessons are the same, and then really think through how to scale up from pilots to more national programs. And then another one of our goals is to make sure that actors that are involved or should be involved in a wastewater-based epidemiology national system, or not necessarily national, but some national as well, that they are brought together. We can use our advocacy capacity to raise awareness, but also to bring these actors that in many countries do not work well together. And this is not a new issue, let's say. And I'll talk a little bit later about where we're doing this right now. And perhaps for, let's say, my final slide, some key points that we always bring in our dialogue. Key points about what are the current challenges and opportunities. And I'm not talking about technical challenges. I think we know, or what they are. yeah, I think the technical aspects are quite well, let's say, mastered at this point. Although there are some, there's still some improvements and research needs not to improve, to keep improving them. But here today, I'm focusing more on the institutional and financing challenges that we see and the opportunities. And one opportunity that we always mention when we discuss with governments is, no, it's not too late to start wastewater testing, because some may say, well, the COVID-19 crisis is coming to an end, entre comillas, no? And we all know that's not quite true yet. And so that's one thing. But the other thing is that we've discussed today already that there are other uses for wastewater-based epidemiology. And so our main message to them, to the governments, is no, it's not too late to start. Actually, it's the right time to start. Today is the right time to start. And as Tatiana mentioned, let's not forget the non-sewered population. So no, and the principle of equality, let's say, making sure that everybody can benefit from what wastewater-based epidemiology can bring to the existing health surveillance system toolkit. The other message that we always bring in our discussions is, so let's start moving from pilots to scale, no? And that brings in a big challenge of multidisciplinarity, making sure that all stakeholders are there, wastewater and water, utilities, health authorities, academia, research centers. But not only do we need to raise awareness and seek leadership from the health sector, but we also need to make sure that the benefits are understood from the ministries of finance. And this is where we've stepped in recently to making sure that the message goes all the way up to the ministries of finance if we're going to have this, if this is going to be sustainable. And this brings me to my third point. Very important that we are able to voice out, to identify, to calculate the benefits for each of these stakeholders, and they will be different. We've had discussions, for example, in Mexico, where the Instituto Nacional de Salud Pública was telling us they need to really raise to the Ministry of Finance, first their own ministry, Ministry of Health, and then to the Ministry of Finance, what are the benefits of doing this? Can it, and if it can, for example, replace part of the national surveys that gather information on the health status of the population, well, how much can we save? That's a very direct, practical way of looking at it, no? Or what does an early warning signal give you in terms of benefits, of costs, or organization, or planning, etc. And I think we really need to sit down and try to quantify as much as possible. It's not always possible, but these benefits for each of the stakeholders, they will be different once again. And we need to follow the decision-making chain, making sure all of the actors along that decision-making chain are comfortable with what the wastewater-based epidemiology can bring for them. And that's not easy. And I think this is where, there's at least where we think we can help. No? And then maybe just to close on this, we're having ongoing discussions with the governments of Peru, Colombia, Mexico, currently, no? And Ecuador. I mentioned Guayaquil. And just to give you some examples, that these examples are completely different. From Peru, we have the water sector that's leading, and they've had some very successful pilots in Lima and Arequipa, for example. And they came to us for support in thinking through their scaling up strategies. And now we're in the step where we need to talk to the health ministry to make sure that they're on board and making sure that there will be resources for this scale up. Whereas, for example, in Mexico, we've received a request from the health sector, and we're more in a position where we need to make sure that the water sector, even though they did pilots through, for example, the National Water Commission, CONAGUA, making sure that they see the benefits for them. And the discussion now is revolving around what if we take wastewater-based epidemiology, but we also increase the borders or the limits of this to water quality monitoring, and really spark the interest of the National Water Commission. So the discussions are different. They're coming from different actors. There's different angles. We need to consolidate these angles and make sure that we can bring these lessons also to other countries we work in. And this is what, for example, we're doing in Colombia. In Colombia, there is interest, but to start with a pilot first. But we need to bring the Ministry of Finance, because we're having discussions on new projects, new investments in Colombia, and we need to convince them that this is a good investment. And so very diverse situations, which just reinforce my point that we need to identify the benefits for each of the stakeholders to make sure that we can scale it up and make it sustainable. And on this, I'll end my presentation, and if you have any questions, happy to answer. And Doug is also here for that. Gracias.
Muchas gracias, Jean Martin. Should I speak English or Spanish?
Para mí el español está bien, pero para Doug creo que mejor en inglés.
Okay. Thank you so much for this very interesting presentation. And for...
We will continue now in English because of Doug. Muchas gracias por esta presentación. Thank you so much for this presentation.
In different countries. I'm sure that many people, including me, are wondering, how could I be part of this? How could I be considered to be part of these pilot studies, or to find or to search for funds to be considered as part of this joint effort?
Very good question, and maybe I can perhaps say explain really quickly how we work. And the reason why my presentation was very focused on government is that our clients, our primary points of entry into countries are the governments. So they usually come to us with requests. But then when we receive these requests, we usually tell them to open the discussion, to other stakeholders, including academia, including research centers, etc. And this is exactly what we want them to understand, is that if we're going to embark on this, there's already actors that are working on this and doing it very well, and so let's bring them on board. And usually the financing goes through governments, but it doesn't mean that it doesn't go back to the other actors after that. But so we play a convening role where we make sure that all voices are heard, particularly if these voices already have experience that is going to be beneficial to let's say a scale-up program. But keeping in touch is a good way for us to make sure that your voices are heard, your experience is shared, and who knows, if there's a project that incorporates wastewater-based epidemiology, there is going to be financing associated with that, and for the involved parties.
Thank you. I will read the first question that was proposed by the audience. It's in Spanish. But I will translate it. So during sequentiation you mentioned that the sample processing could be done in the same place that is obtained. What factors could affect the processing in the moment or after that moment, or after taking the sample? And what guarantee of having an innocuous sample? Does it make sense?
I'm not sure I understand. Is it about?
Also for you.
Okay, what is it in Spanish? to make sure.
Durante la secuenciación mencionaba que el
During the
proceso
sequencing, you mentioned that the processing of the samples may be performed in the same place where they are obtained. Which factors might affect the processing at the moment of collecting the sample or later? How can we have a safe sample? I really don't know if this question is for me because I didn't talk about this topic. I am sorry, I didn't talk about sequencing. I don't know, Doug, do you have any insight on that? Or maybe Tatiana?
previous speaker probably. So I will move on to the next question. Yeah. Have you done economical study about the cost benefit of implementing systems for environmental monitoring?
And that's exactly where we want to, that's the work that we're doing right now. This is our next step because of the discussions that we've had with ministries of finance and ministries of health. They're asking us, how much does it cost? Is it worth it for me to invest in this while I know that there's still people without a connection to a sanitation system, or drinking water, etc. So we need to contrast these, and that's the work that we're doing right now, is to make sure that there are somewhat clear numbers that can be associated with environmental surveillance. For example, taking the costs from Europe, from the US, Canada, and our pilots in Latin America, we estimated that it would be let's say up to $1 per person per year to set up the environmental surveillance system at scale, and then the cost would be reduced to, let's say, between 30 cents to 50 cents a person a year. It's very cost effective, but we still have to cost these and make sure that we have a discussion around them, because these numbers, let's say if it adds up to $10 million, maybe 10 million is a lot if there are other competing priorities. But yes, we're working on this, and we'd be happy to share this work once we're done.
Thank you. Thank you,
Jean-Martin. Or maybe I'll just add one point too, is that we had a lot of discussions about costing, and one of the challenges are that the cost can vary quite a bit. If you have a city of 1 million people, that can cost just as much as a town of 10,000 people in some ways. So I think, when you think of that, the formulas for that, I think there's going to be a point where we need to have a discussion collectively as a community about what these parameters are. And then as countries need to roll out this or interested in it, it's going to be country specific information that would kind of, I think, could go into a template. But one challenge of these sort of dollar per person per day is it does vary tremendously. And then of course, we want these to reach hard to reach communities where the costs are going to be higher, or there may not be as much coverage with formal sewage systems. So that balance as well.
In terms of that, Doc, or Jean-Martin, your analysis of cost benefits is only for SARS-CoV-2 or COVID-19, or you're thinking to complement to other pathogens that may get much more impact and download the cost of the analysis?
The very broad figures I just shared were for SARS-CoV-2 only, but this is part of our report, for example, that we published in January. But now that we've had discussions with governments, many governments in Latin America, we realized that we need to go beyond that, and of course, and our, let's say that the next steps for us is to calculate the benefits associated with not just COVID-19, but other targets. We haven't quite started yet. We're really, conceptualizing it at this point, but we received really clear requests from these ministries telling us, I need some numbers, or I need something to be able to mobilize financing for this.
In the report too, we highlighted the pace of innovation that's happening. So at the beginning of the pandemic, a $10,000 auto sampler, and now you can have a 3D printed sampler that can work maybe just as well in many, if not most locations. And the same thing, like we heard earlier, today, multiplex approaches, or instead of going one organism at a time, I think we will be looking at multiple organism technology. So even the auto sampler, $10,000 to $30, in a year. So I think we are seeing a rapid decline in cost in terms of the technical parts. And then I think for Latin American, Caribbean too, we've talked about the World Bank. Is there the potential for scaling across countries, like information systems or methodologies, common elements that every country needs to lift up, and can those be templated or done across more than one country? I know that Panacea has been doing more of the work than anyone in that area.
Thank you.
Well, thank you very much. I think we can move to the next presentation because we kind of ran over time.
Gracias, Verónica. Nos queda la última presentación que la va a realizar.
Thank you, Verónica. We have the last presentation with David Graham. David is a professor in ecosystem engineering of the University of Newcastle in the United Kingdom. His group combines engineering, microbiology, ecology, mathematics, biochemistry, molecular biology, process in relation with One Health, including the treatment of residuals, and also the environmental impact of the genetic trace, also using the markers of wastewater to face the pandemic. David is a very important collaborator of ours. Also, we worked with him in Paraguay.
We are very interested in your presentation, that this environmental dimension of antimicrobial resistance under One Health agenda. Thank you very much, David, and welcome.
Thank you, Pablo, for the kind introduction. And also the organizers of this workshop. It's really been very interesting. I'm going to talk about AMR and wastewater epidemiology from a very big picture perspective. And I'm trying, the purpose of this talk is to convince people that don't believe the environment is a very important player in health protection, and monitoring it the way we are right now and what we've learned through COVID might be a way of addressing other big problems like antimicrobial resistance. I'm going to start, and there's going to be a little bit of overlap between my talk and Professor Gonzalez's talk. I want to go back and talk a little bit about what antimicrobial resistance is, but why it's been so hard to convince people that the environmental element of AMR is so important. But I'm a wastewater engineer, and I'm interested in solving the problem. So gathering the data is part of the process, but solving the problem is what our real goal is. And I see wastewater epidemiology as the evolution and recognition of its existence. I mean, we've been doing this for 30 years as the stepping stone that we need to actually start addressing the global increases in AMR. So I'm going to talk a little bit about the process. I'm going to talk a little bit, I'm just only going to provide two fairly simple examples, and then I'm going to talk about solutions, which I find which I feel is really what we should be thinking about at the end of the day of this session. So, to remind you, and this may be completely obvious to 95% of the audience, but resistance is the capacity of an organism to defend itself against an antibiotic or antimicrobial agent. Now, as Professor Gonzalez says, there's two types of resistance. One is called intrinsic resistance, and that's the resistance that exists within organisms in nature and has existed for hundreds of thousands of years. So it's what's in the background. It's always there. What we've done by using antibiotics and, for all intents and purposes, polluting our world, we've accelerated bacterial evolution to create much greater pool of organisms that are resistant to antibiotics and antimicrobials. And this is called acquired resistance. And so what we were actually trying to do, and in the process of increasing this pool of acquired resistance, we've actually increased the pool of intrinsic resistance. So the question is, how do we go back to a world where we haven't created all this resistance? And I don't think it's really possible, but at least we can slow it down. And so one of the things that I've learned in the years that I've been doing work on AMR is that resistance predominantly occurs in the gut of the animal or the humans that's actually taking some sort of therapeutic drug. The problem is that when you defecate, those resistant organisms and genes enter the environment. And unless you manage those wastes appropriately, and this is true in agriculture and in human systems, you then are starting to spread resistance and the genetic potential resistance, which then is amplified by all those things in the environment that we're also putting there: chemicals, metals, other bacteria, zoonotic pathogens. This whole suite of things, the environment is like a big mixing bowl. And in fact, that's really part of the problem. Now, I want to make one really important point before I even start talking about other things. It's been a big deal in the last little while about AMR being the other pandemic compared to COVID. But in fact, we've known about the AMR pandemic for quite a few years. And I show you just one example here from work Peter Hawkey did at the University of Birmingham. And he tracked back the history of extended beta-lactamase resistance of one particular gene class called CTXM, which was first detected in the environment in Eastern Europe in an animal farm. But in 5 years, this particular form of resistance was now prevalent around the world. And there was many different variants that some of which were much different than the original, but also most of which were much more serious relative to having a pathogen acquire it and then it be able to defend itself relative to therapeutic treatment. And so what we basically have observed in 1989 to 1994 was exactly what happened with our COVID, with the COVID proliferation. Now, this cartoon that I'm showing you is what happened over about 9 months relative to COVID, but we think this is happening simultaneously with all the different other types of resistance genes. Now, we typically in our assays quantify about 400 different genes. So each one of those genes is going to have its own story relative to the evolution of how it spreads, which predominantly we believe is in the environment, and then how it's going to evolve and mutate and become something probably worse. So if we go back to the origin of why AMR hasn't been addressed to the extent that we feel it should, is because I think AMR is trickier than SARS-CoV-2 as something to try and mitigate, solve the problem of. And I want to go back to the very basic and simple way of looking at what actually happens in the evolution of AMR at the bacterial or microbial scale. So you got this bacteria or microorganism out there and you treat it with a chemical, which we call an antibiotic. Now that's going to kill most of the bacteria because most are susceptible, but there's going to be a small few based on that intrinsic resistance that we know about that are going to survive. Those are not going to have any competitors anymore, and ecological selection will cause the resistant strains to become dominant and thrive. So this is the way resistance evolves and why we are finding more resistance. Our problem from an environmental management perspective and health protection perspective is this is happening simultaneously everywhere we're doing something that's related to an antibiotic or even pollution. We're using antibiotics in humans, we're using it in animals, we're spreading chemicals into our crops, we're polluting our water with fecal matter. And that's happening everywhere simultaneously. So when I, as an environmental engineer, am going to someone in the medical profession and I'm trying to say the environment is absolutely critical, well, they will ask me, well, show me a pathway. Well, the problem is that there's so many different pathways that are overlapping that it's really hard to define each individual pathway and then provide the advice to the health professional that the health professional needs to act upon the information that you're providing them. So we have all these things happening simultaneously, and the environment's the mediator. So from a mitigation perspective, which is my goal, is we must identify the critical sources. We must reduce those sources, and sources, I mean sources of AMR genes, bacteria, or anything that will promulgate the evolution of new resistance. And in what resistance exists, we need to slow those pathways. Now, I'm going to show you a figure that I absolutely love. I was involved in writing a report for the World Economic Forum that was released in last August. And I think this report is really good. It's a little mini version of the O'Neill report, for those of you that know what the O'Neill report is. And what we basically wanted to do is look at the relationship between water quality and the spread and evolution of AMR. So first we mapped out the sanitation levels around the world. And this is a slightly incomplete map, but it's a pretty good map, defining the relative level of sanitation on a global scale based on the relative level of wash infrastructure. So the key thing for looking at this map is, I'm going to change this map. I'm going to overlay on top of this map what we've estimated to be the risk of exposure of AMR in 2030. So the key thing is to look at the change in the colors of different parts of the world. And some of the colors in Latin America change. Some become darker, which is the risk is higher, and some actually get a little bit lighter. But some parts of the world get very, very dark. And so you can see that in Latin America, Brazil has got darker. In Asia, China's got darker. India's got darker. And these places have got higher risk because of the non-sanitation factors that are prevalent in those places. It might be in one country, it might be high levels of antimicrobial manufacturing. Another place, it might be high levels of use of growth promoters in animal rearing and in animal agriculture. The bottom line is that these changes in colors and this increased risk is what I call the one health factor. In other words, sanitation by itself can explain risk, but the amplification and increasing levels of real risk relative to AMR is the overlapping of all these things on top of each other. So this is the projected conditions in 2030 accounting for climate change in terms of the modeling. But if we look just at this little cartoon in the top left, there's two parts of the world based on monitoring, sewage monitoring done about 4 years ago by a group in Denmark. And it's a fabulous, fabulous piece of work. Two parts of the world have got much higher levels of AMR per capita in their sewage than other parts of the world. And those are in Africa, where the highest levels exist, and the second is in South America. But it's interesting that they're higher, but the error bar is also bigger. And the reason why the error bar is so big is because some parts of these continents, there's perfectly satisfactory waste management. But in other parts of the continent, there is all but none. And so one of our critical goals, and the last speaker I thought said it very well, is that we've got to make sure that when we start using monitoring and wastewater analysis, that we're actually capturing everybody, not just the people that live in the big cities that are near wastewater treatment plants. So the thing that's really noteworthy here is the places with the highest levels of water and wastewater infrastructure have got the lowest levels of AMR. And that's not a mystery. That's because they have better quality water. They got better quality environments. And one of our goals is really to have that aspiration for the whole world. But the question is, how are we going to do it? So that's where wastewater epidemiology becomes important. We know AMR development, transmission, and spread is increasing everywhere, but we want to try and mitigate it. We want to somehow come up with strategies that allow us to actually reduce this spread. Ideally, it would be stop it and have it turn back, but I'm not optimistic that's going to happen. It could happen. But wastewater monitoring and water monitoring is the first and critical step we have to do to be able to turn the AMR evolution and transmission problem backwards. Wastewater monitoring can identify places that require immediate action. But this immediate action varies from place to place, and I'll give you a couple of examples in a moment. It can guide human animal health protection strategies, including the importance of improving environmental quality, which then in fact improves health and reduces the requirement for potential antibiotics. So it's an upward spiral rather than a downward spiral. Finally, wastewater monitoring can source track drivers of local and global AMR spread. In other words, I showed you that figure before of all these happening over top of each other. But by genetic source tracking, and we've done some of this ourselves, is we can identify the particular places that are the main drivers of resistance in a particular location. So I'm just going to give you two examples, and the two examples are actually very simple. One is from 15 years ago, and it was a study that we did with Susana Olivares and her team at the University of Havana and INSTEC in Havana, in Cuba. And the second case is something we've just done a couple of months ago in the UK. This original study, so what we did was we looked at AMR along the Almendares River, which is a river that's on the western edge of Havana. And I provide a map here. And what it's got is a very mixed land use. There's suburban areas, there's a zoo, there's factories, there's a wastewater treatment plant, a landfill, and then there's mixed urban development. And originally this study started as being a study on heavy metal pollution. And we did that work, and it was done by Susana's group in 2004 and 2005. We got involved a few years later, and it struck me because metals can exacerbate AMR evolution, increase it. We were curious because of the high levels of metal pollution in the river, we might be able to detect something equivalent, that it might mirror something to do with antimicrobial resistance. So we monitored the river. We also monitored the sewers in the vicinity of the river. And this is what we found. Bear in mind, this was before the time we could do massive sequencing, but we did qPCR using about 30 different genetic targets. I'm only going to highlight 3 for the sake of my example here. The top graph is beta-lactam resistance, which is antibiotics like amoxicillin. The middle graph is erythromycin, and the bottom graph is tetracycline. And I think the first thing that I hope is obvious is if you look at the 3 different classes of resistance, they show different patterns along the river. So what we're seeing already is AMR is not a single thing. And it's a whole series of different things that are going to be driven by different factors. And that's why I've said that AMR, I think, is a trickier problem than SARS, because we're looking at many different many different targets simultaneously. But when we actually got the data, we stepped back and said, okay, let's think about the catchments and the landscapes and how can we solve this problem? And so what we did was we systematically went through each step and identified the main things that were happening in the catchment that were causing the different peaks that we were seeing in the AMR mosaic along the river. And these were our conclusions. In the upper part of the river, we saw this hump of elevated levels of resistance of all 3 types of resistance. The problem was that we couldn't find any source. And a few years after we did this work, I had one of my master's students go to Havana, and she studied this problem, and she discovered the problem was septic tanks that weren't being maintained in the ground. And in fact, they were leaking into the groundwater, and the AMR was coming up from below, from the groundwater into the river and the headwater of the river. When we went downstream, we saw this incredible peak that was below where there was an outfall point where there was industrial waste entering the river. And those industrial wastes included waste from the pharmaceutical industry. And we also found there was high levels of amoxicillin. Finally, at the bottom of the river, we found this great elevation of tetracycline resistance. Now, initially, it wasn't completely obvious what this was, but then we started thinking, okay, well, how could you get tetracycline resistant when there was no tetracycline there, really? And what we subsequently learned was that heavy metal resistance, which is in bacteria, also the gene cassettes that carry the metal resistance genes are often also gene cassettes that carry tetracycline resistance genes. So what we think this bottom peak is related to heavy metal pollution from the landfill. Now, the good news is, and this is the reason why I use this example, even though the methods are old-fashioned, is it actually led to 2 solutions. And the Cuban government took to heart the results and and they put in wastewater leachate treatment at the landfill, and then they directed the landfill effluents away from the river. So they reduced the source and they directed it away from the water where people were. Relative to the industrial waste, there's been much more stringent control on waste releases, which is tremendously good, and it should be a model for anybody else elsewhere in the world. To my knowledge, I don't know whether anything's happened related to the domestic waste, but we've seen it. Now that we've learned about this possibility of leaking septic tanks and AMR entering a water source from the groundwater rather than the surface water, it's led us to study this in many other countries, and we've learned from it. But we ultimately ended up with a suite of solutions from this type of monitoring. The other example is more just, and this sort of just restates something that Professor Gonzalez said, is we've recently been monitoring using multiplex qPCR assays, which Professor Engelin had talked about, where we're monitoring about 400 genes simultaneously using a qPCR array. And what we were curious about was just if you chose sites across the community, could you see sources that could explain elevated level of AMR at those sites? So we did bias our sampling a little bit, but this associates with this array of different sites across the top of my graph, which goes from C1 to C12, but there's 4 sites that are H1, H2, H3, and H4. And those are the sites, these are some in-sewer sampling, and those are the sites that were close or immediately downstream from hospitals. Now, I'm not indicting hospitals relative to their waste, so it's something that we can talk about. But what this shows you is just by doing the multiplex qPCR array, looking at the landscape, you can actually come up with information that can then guide policy and strategic decisions relative to healthcare, and in fact, this potential spread of AMR. So, this is my second last slide. And so, as I said, my goal in all of this is to try and come up with solutions. And so we've been studying AMR in many countries for quite a few years, including Latin America. Solutions to something like AMR, One Health solutions, really have to be a combination of things. It can't be just one thing. It's got to be social and behavioral, and it's also got to be followed with technical solutions. Social behavioral, I mean, just altering the way we use antibiotics. Use antibiotics prudently. We cannot not use antibiotics, but we need to be more conscious of how and why and where we use them. But there's other things that become really important, maybe particularly important in a place like Latin America, is when it comes to waste releases, if there's rules, they've got to be enforced. If you've got infrastructure, even if this infrastructure is imperfect, maintain it as well as you can. And finally, I think we globally have to empower local action. And that's what I really appreciate about some of the comments from some of the other speakers. And also the whole purpose of Panacea as a working group is it's a way of empowering action and putting the power into the people that actually know their places and understand their problems. Now, to be very honest, I, as an engineer, have a technical way of solving almost any AMR problem, developing infrastructure, improving sanitation, putting in extremely sophisticated wastewater treatment or water treatment. But the thing is that doing that is just simply too expensive, at least in the first instance. And I think then this is where I get back to the environmental dimension being most exemplified by One Health solutions, is you've got to combine things like changing behavior with increasing technology, but you don't have to have the highest technology to have a really big positive effect relative to the levels of resistance in the environment. An example being is just implementing water sanitation and health hygiene. It has an incredibly big effect relative to the spread of AMR in locations where it's implemented. So, in my final thoughts are, reducing global AMR will only be addressed if we think in a One Health way. So we have to think holistically. Environmental factors appear to dominate the spread of AMR in the world. we may use antibiotics, and they may cause AMR to evolve and develop, but its spread, this global movement, is driven by factors to do with the environment. And I think we have to convince people who make decisions at the highest level that this is critical. With that, the best way we can start coming up with solutions is by fully implemented wastewater epidemiological approaches. We can monitor it, they can tell us the prevalence, the types of AMR, and then that can help us design the best ways of solving the problems on relatively local scales. And small solutions can have a big impact, just for example, of implementing WASH. So the way to address AMR is to implement AMR wastewater monitoring on a global scale. But that central to that is increasing regional capacity with common methods. And that's what I think is so great about Panacea. Thank you.
Thank you, David. I will check if there are some questions from the audience. But I think that is quite interesting, this difference between, you found in the previous work that you've shown that in Africa and South America, it's a quite different, it's a standard deviation now. And it's because of different realities of different countries. In terms of that, wastewater epidemiology from treatment plants could be easier for big cities, but in countries like us, like little countries like Paraguay, where we don't have many treatment plants. The cost of doing this analysis in rivers or small towns is increase a lot of the cost of this. So how to approach this reality to make a wastewater surveillance in this kind of situations?
Yeah, and that's a really, really good question. And the way that we've been approaching this is first, there's a sort of a two-part question. The question is, if you don't have an obvious infrastructure from which to sample, where do you sample? And how do you gather something that mirrors the community in an adequate way so that you can actually provide realistic advice? And in fact, we're working on that. And we're working with colleagues in other countries on that. And you and I have talked about that. But it's we've come up, I won't give a lecture on this. I'm an old professor, so I have a tendency to lecture, but we've got 9 scenarios for monitoring that mirror different types of circumstances where you might be able to gather information about a particular place, ranging from sampling a place where there's functionally no sanitation, which might be a ditch by the road, versus a pit latrine, versus a septic tank, versus, there's different levels. But the question is, this is the thing that we've been working on in the UK, is trying to say how, if you get a sample from a place, how representative of that place is the sample you collect? And when you don't have a collective system of the waste combining, then you have problems coming up with, what is going to mirror the real system versus what you collect in an individual sample. And I don't have a good answer, but I would say it's something we're trying to think about. In the UK, the rule of thumb is we want to capture about 10,000 people within a reflected sample. And that would be the optimal. Now, that's actually sub-waste treatment plant levels. So you wouldn't be sampling at wastewater treatment plants. You might be sampling in line in sewers. And I think I sort of view the same thing, except you might have to sample a little bit more when you don't have an infrastructure. So that gets to the second part of your question. The second part is, well, how can you afford it when you've got this multiplex qPCR or Illumina sequencing? Well, to be honest, you don't have to use those methods. There are other ways of gathering information that are considerably less expensive, but give you a really good reflection of whether there is AMR in a particular point in space and time. And I'm really kind of old-fashioned. We measure. Even if we're studying in the most developed or least developed country, we always do E. coli, culture E. coli, and we culture ESBL E. coli. It's what we do. And it's what the World Health Organization is recommending as well, as at a very minimum. But if you could do that for your local, I'm not talking you personally, Pablo, but I'm saying if one can do that in their own area, we can provide you some really good advice. In other words, that's enough. Now, the next step is, okay, if we have some genetic capacity, and that's one of the benefits of having just gone through or in the middle of a pandemic, is that the world has become much more genetically competent. So we could step into, say, qPCR methods, not multiplex qPCR, just qPCR. Well, we've now monitored, not we, but the collective community studying the environment have now studied thousands and thousands and thousands of samples. And I could probably tell you 5 things to do qPCR on, and you get a pretty good idea. You get 80% of the information you need to make a decision about waste AMR management. And so this is something we're doing. Another thing we're doing right now is, okay, do we need 3? Do we need 4? Do we need 5? Which of the ones do we want? So that's the next level of surveillance. So what I would do is I would do culturing if that's your capacity. The next thing would be culturing plus selected qPCR for sentinel markers. And then the next step would be up to multiplex qPCR, and ultimately you would be doing the sequencing that the group at Karolinska can do, which is incredibly impressive. But the thing is, that's part of what I was meaning about think small. But there's a lot of very valuable things we can learn from little things, little actions. And so I know I just gave a whole talk based on your one question, but it's something obviously I'm kind of passionate about. And but I think it's doable. But you've got to, we can't do everything at once. We've got to be progressive and take that approach to the solutions.
Yeah. I think it's one of the challenges of Panacea, try to understand these different complex situations that we got in South America and Latin America.
Yeah.
And that this is quite important. Verónica, te dejo la siguiente pregunta.
Okay, thank you. It was a super nice presentation, but and thank you for answering the previous question. It was very, very, very good too. So this is a question from the audience. So which could be the first strategies or tool to apply for AMR monitoring in wastewater in a country of in a low-income country?
So what would be the first tool?
The first strategies or tools to apply for...
No, I think I sort of answered the question, is that I would first you want to see what the internal capacity of the low to middle income country is. But most places now have got the capacity to do microbial culturing and qPCR. So we could say, I could tell you 5 or 6 specific things you would do qPCR for. I would couple it with culturing. So that's what I would do first. And it's because of the fact that genetic capacity has increased in many, many places that we can be that ambitious. So people think that those older ways or the less fancy ways aren't as useful, but they are. Coliforms are in your gut. E. coli is in your gut. If it's drug resistant, it means there's drug resistance in your gut.
David, it's quite amazing. I'm reading the question from the audience, and you approach it in the first question, most of it. But so that this is, I think everyone here in Latin America had more the same question about it, how to approach this problem. It's quite amazing. But what is the perspective do you have regarding the industrial waste? How to manage the situations?
It's one of the trickiest ones because I know in Latin America, industrial waste pollution is quite a big problem, especially its legacy pollution. And I think some industrial waste, I mean, if we go back to think of what causes AMR is caused by stressed microorganisms. It doesn't have to be by an antibiotic. It's just got to be something. I mean, part of the stuff we were doing on the Almendares in Cuba was by a lead smelter. Now, lead is very toxic to microorganisms, and we found an astoundingly large number of antibiotic-resistant organisms near the smelter. that where there was no antibiotics at all. There were none. It was just based on co-selection based on the metals. So it's a hard question to answer, but this gets sort of back to what I said about enforcing rules. Now, every country in the world has got legislation about metal pollution or all these other pollutants, and these are the types of things that come out of industry. And if you enforce the rules and the industrial wastes are treated, which is perfectly possible, I could probably, every industrial source, I could come up with 10 solutions to reduce the releases. But it's a matter of having the political will. And I think that's maybe the dilemma, is that many places are very dependent upon the industrial activity for jobs and health, well-being, but they're paying a price for it relative to their health. So it's a great question. And the answer, unfortunately, is a political answer. It's not a technical answer. We can solve it technically. It's just a matter of doing it.
Thank you, David. I think we are on time, so this is the last question, but it's quite interesting from your previous one. That is, what is the risk of perform old-fashioned method in a next generation sequencing world? Would we increase technological gap? Would we increase that analysis capacity gap?
Oh, that's an excellent question. The answer is the goal is not to have the old-fashioned method be a substitute for the new method. So what we want to do is, the way we do things is we started off 25 or 30 years ago doing E. coli and with culturing methods. Then we developed qPCR methods, then we developed multiplex qPCR methods, and then we did sequencing. We still do culturing, but we also do sequencing. And I think the goal is to gradually progress a richer array of methods rather than substitute methods. It's because ultimately, if I'm trying to talk to a physician and convince them that there's something in the water that's going to cause them a clinical problem, they don't care about whether there's a gene there. They care whether there's a carbapenem-resistant Klebsiella. And that's what you can learn from the culturing and the more simple methods. So you can actually provide more immediately, at least superficially useful information for somebody who's making a healthcare decision by including the simpler methods within the array of your growing skill set, which ultimately would be sequencing. So I think I definitely would not want to have a technology gap, because that's not the purpose. It's to extend and make the capacities richer and more versatile in any place that might want to do monitoring.
Thank you very much, David. Sorry, Veronica.
I just wanted to thank David. It was very kind of you to answer all our questions, many questions.
Well, thank you for inviting me.
I think
that
we
was very
need
nice.
to move to the conclusions. Otherwise, we will run out of time. Voy a empezar a hablar en español.
I will switch into Spanish now. Pablo, Aldo, and Maria Inés. We are going to now start with the conclusions in order to be able to close this event. Well then, everybody's in silence now. Now I would like to make some remarks. I believe that all the presentations have been quite interesting this morning, and I would like to underscore a few items that I will mention to see if you agree with me or if you could add others that I have not taken into account. According to me, it has been very interesting to hear the experience of different labs and institutions related to the work of reconversion in order to solve the problem posed by the pandemic urgently. So we have seen that lab cases, and to put the analysis capacity at the service of public health, how to provide more faster information. And to that end, we have also heard the contributions made by Tatiana. We have heard as well the interface of communication once the health authorities are contacted. And they also mentioned, David talked about the other pandemic, the antibiotic sequencing, and they also described, as stated by Bruno, and it was also very interesting to hear about all these experiences of the World Bank with different countries to approach this problem. These are more or less the most outstanding points which, according to me, have been introduced today. Of course, with a lot of valuable and interesting information. Would any of you like to add anything? Well, I thought it was very interesting to resistance and a lot of SARS-CoV-2. I mean, how can we see them as compatible? See wastewater cases related to only one pathogen could be considered to be most costly. This is a very important source of information. I love the presentations by Tatiana, David, and the World Bank on this. And it was also very interesting to hear, I don't know if it was Jean Martin or Doug, who said that we have to understand this as a multidisciplinary idea. I mean, we need to have water, utilities, health officers, the academia. I mean, we need to have a multidisciplinary approach. And as David said in the end, and this is the interest of Panacea, and I think that this will be discussed tomorrow when each country starts to tell its own story, the different realities that exist in South America and in Latin America, throughout Latin America, on how to approach the problem. And this is something that is food for thought. I mean, these are a few of the ideas that were covered. Thank you. No doubt, I totally agree with the comments made by Veronica and Paulo. This is something that we have to work hard on in our Latin American countries. As Tatiana stated, and now Pablo, we need to sit down with different instances. In the academia, we see the need to exchange ideas with the different ministries. In the specific case of my country, there is no coordination, as I will explain tomorrow. I mean, we generate data, but then these data are not really useful. Decisions are made based on those data in the specific case of SARS-CoV-2, but this could be extended to run data and others. I mean, these are very, very important. And the other very relevant thing, the quality of the presentations this morning is extraordinary speakers. And also it is relevant to see the unbelievable contribution of information which may be generated by 1 liter of wastewaters. We have seen some details, but there are many other data that we were able to find in wastewaters. I think that this would be my conclusions for the moment. Maria Inés, would you like to add anything? I agree with all of you. I think that this was a very fruitful day. We had very rich presentations. We have excellent information to work in the future through the Panacea network. I think that one of the most essential problems is to convince the other countries to organize based on this core problem, because I believe that in Latin America we all want to make those things, but we have, despite of that, very fragmented information. The challenge is how it is that we have to work with. I have worked with polio, environmental surveillance with polio, and we have a guidance of the WHO for environmental surveillance. This works very good in all the different spheres or arenas of globally. This is a topic that we have to start working with in the agenda, because there are many countries in Latin America that are at a very high risk for the reintroduction of polio because of the low vaccination rate. So we really need to find now to discuss how it is that we can get organized to consolidate this in Latin America, because we have great possibilities in epidemiology and to face what we are now seeing. This has been done for polio, for microbian resistance of something which is not very complicated. As Aldo said, it's only having a liter of wastewater can help us with so much information. This was a very good day to consolidate the information brought throughout this network.
Muchas gracias.
Well, I would like to thank you all, Pablo, Maria Inés, Aldo. I would like to thank all the different speakers. Exceptional presentations, a very fruitful day, very interesting. And of course, I would also like to thank all the people who joined us through YouTube. I adjourn the work for today with such rich interaction. And I would like to invite you to join tomorrow this workshop at the same time in the same channel. Thank you so much. Have a good day, good evening, good afternoon, depending on where you have logged in. Thank you so much and see you tomorrow. Thank you.
Listo. Muchas gracias a todos. Ya se